ये पाठवलं गुरु वॅक्सिनेशन.
हॅलो हॅलो सर.
हा हॅलो सर.
थोडं लॉग आउट झालंय टेक्निकली, होतो लॉग इन पुन्हा एकदा. ठीक आहे,
ओके ओके सर, काही प्रॉब्लेम नाही.
लॉग इन होते पुन्हा एकदा.
हो हो हो, काही हरकत नाही.
हॅलो सर, टेक्निकल थोडं लॉग आउट झालेलं आहे. लॉग इन
काही
होतो
काही
नंतर पुन्हा एकदा.
हा, काही हरकत नाही सर, काही हरकत नाही.
फॉर 1 मिनिट.
हो हो हो.
Okay. Good morning to all. Today we are going to study parametric test, that is t-test, and whatever numerical problems are there, they also will be studied in this our lecture. So if any problem or doubts are there regarding earlier period, also we will discuss at the end. So now I am going to share the screen. Okay. Hello. Is it visible the screen?
Yes, sir.
Yes, sir. Yes, sir.
Okay, thank you. T-test is a parametric test. In last period or in last session, we have discussed about the chi-square test. It was non-parametric test because there were no any parameters for applying the test. But here we need to follow some parameters of the parent population. That's why T-test is considered as parametric test. First, the theory part. T-test is a statistical test that is used to compare the means of two groups. Means means averages. Means means what? Averages, arithmetic means. It is often used in hypothesis testing to determine whether a process or treatment actually has an effect on the population of interest or whether two groups are different from one another. Means here we are going to test the means in between two groups or in between sample mean and population mean also. The t-test is a parametric test of difference, meaning that it makes the same assumption about your data as other parametric tests. The t-test assumes your data. यामध्ये आपण assume करता, काय assume करता? Your data are independent. Means whatever data we are going to collect, that is considered as an independent data. Then data are approximately normal distribution, means it is expected their normal distribution is there, symmetrical distribution is there. Then number 3, have a similar amount of variance within each group being 1 minute. Okay. It means what? Whatever variance is there, variance means nothing but it is the square of the standard deviation. It assumes that the data have a similar amount of variance within each group being compared. When choosing a t-test, you will need to consider two things: whether the groups are being compared come from a single population or two different populations, and whether you want to test the difference in a specific direction also. या ठिकाणी under he test we are going to compare the mean of one sample with another sample, mean of sample with the population mean also. On the basis of that, t-test can be classified as first one sample test, second one two sample tests, or third one pair t-test. First one, if the groups come from a single population, that is measuring before and after an experimental treatment, perform a paired t-test. Paired t-test whatever your sample is there, that is the same and from the same population, but you are taking the measures or you are taking the observations before experiment and after experiment, or before treatment and after treatment. Likewise, means sample is remain same, but we are taking the measurement before and after. That is called as a paired t-test. Paired t-test means we are taken into account the mean of before and the mean of after, and then will be compared. Second one, if the groups come from two different populations, for example, two different species or people from two separate cities, perform a two sample t-test, that is independent t-test. Here, whatever two samples are there, these two are from different populations, not from same. It means that we are going to compare here the means of one sample from one population to another sample of another population. That is called as two sample test. Number three, if there is one group being compared against a standard value, perform one sample test. Means where there standard value means what? Standard value means population, whatever mean is there. Population or hypothetical mean is there, that is called as standard value. And from that population, when you are taking one sample means sample whatever size is there, and the mean of that sample will be compared with the population mean or hypothetical mean, or I mean standard value of the population. That is called as one sample test is there. Okay. Next one. To test the significance of the mean of a random sample, first category, here we are going to test the significance of mean of a random sample. कुठल्या एका random sampleचं mean आपण त्याच्या population mean सोबत parameter सोबत या ठिकाणी टेस्ट करतो. T-test is used to determine whether the mean of sample drawn from a normal population deviates significantly from a stated value or standard value or hypothetical value or population mean or value when variance of the population is unknown. In case the variance is unknown of the population, in such case, whatever standard value, whatever hypothetical value, or whatever I mean population mean is there, that is compared with the sample mean. That is a one sample test. That is what? One sample test and nothing else. Means here, the mean of one sample will be compared with the mean of the population or hypothetical mean or value. The formula is for one sample test is T is equal to T, small t is the T value, T test value. Firstly, X bar means nothing but whatever sample data is there, for that sample data to take the arithmetic mean is called as X bar. Then minus mu. Mu means what? It is the standard value or hypothetical value, or I mean population mean. First is the sample mean, X bar is the sample mean, and mu is the population mean. Then under root n. N means nothing but size of the sample. Number of observations under your sample is considered as n, and divided by s. S means standard deviation. Standard deviation means nothing but we know already, But here, in case of standard deviation, we have to divide by whatever squared deviations are there, which are taken as a sum, divided by degrees of freedom, means n minus 1. So here, x bar is the mean of the sample, mu is the actual or hypothetical mean of the population, n is the sample size, s is the standard deviation of the sample. Now, second category, to test the difference between means of two samples independent, means suppose there are two different populations and or same populations, but your samples are two. Your samples are two different. And in case of two sample test, we have to firstly find out the means for two samples separately, mean 1 and mean 2. And then we are going to compare the mean of one sample with the mean of another sample. That is called as two sample test and nothing else. If there are given two independent random samples of size n1, means size may be different. First sample may be of 8 observation, second sample may be of 12 samples or 7 samples, maybe. So size of n1 means the size of first sample and n2 means the size of second sample, with the means of x bar 1 means mean of the first sample, x bar 2 means mean of the second sample, standard deviation s1 means first sample, standard deviation s2 means second sample standard deviation. Then we are very interested in testing the hypothesis that these two samples come from the same normal population or not, applying t-test. Here t is equal to अगोदरच्या एक्झाम मध्ये वन सॅम्पल टेस्ट होती, त्यामुळे एकच सॅम्पल मीन आलं. बट हियर टू सॅम्पल्स आर हियर, सो टू मीन्स विल बी देअर. So take into account t is equal to x bar 1 means mean of the first sample minus x bar 2 means means of the second sample divided by s means combined standard deviation into under root n1 means size of the first sample into n2 means size of second sample divided by n1 means size of first sample plus n2 means size of second sample. Okay, so x bar 1 we know, x bar 2 we know, then n1 we know, n2 we know. S means the combined standard deviation, not s1 and not s2. It is combined standard deviation of two samples and it is calculated like, okay, yes, or combined standard deviation is equal to, take into account again here, firstly standard deviation we know already. When we are taking the deviations from actual mean of each observation and then squared, and sum of the squared deviation is taken into account here. So in first part, sum of the x1 minus x1 bar whole square plus for second sample sum of x2 minus x2 bar square divided by degrees of freedom means n1 first sample size plus n2 second sample size due to having double n minus not 1 minus 2. So under t-test degrees of freedom is equal to n minus 1, but here two samples are there. So n1 plus n2, due to two samples we have to deduct minus 2 and nothing else. Okay, thank you. This is the theory part of the t-test. Here one is a one sample test and another is a two sample test. We are going to apply practically with the help of the problem. So I am going to share another P1. Okay, is it visible the screen?
Yes, sir.
Okay, okay. Okay. Problem number 1 on t-test. Problem is the manufacturer of a certain make of electric bulbs claims that his bulbs have a mean life of 25 months with a standard deviation of 5 months. A random samples of 6 such bulbs give the following values. या ठिकाणी काय म्हटलं, एक मॅन्युफॅक्चरर आहे, सर्टेन मिक म्हणजे एखादी कंपनी आहे इलेक्ट्रिक बल्बची, आणि त्यांनी क्लेम केला, दावा केला मार्केटमध्ये, काय केला, हिज बल्ब्स म्हणजे त्यांची जी काही बल्ब्स आहेत हॅव अ मीन लाईफ ऑफ 25 मंथ्स. आमचे बल्ब हे साधारणतः 25 महिने चालतील सर्वसामान्य, सरासरी, मीन म्हणजे सरासरी, आणि त्यांचं स्टँडर्ड डेव्हिएशन आहे 5 मंथ्स, म्हणजे 5 महिने पुढे जाईल किंवा 5 महिने कमी जास्त मागे येईल, असं हे हायपोथेटिकल आहे. तर त्या ठिकाणी काय म्हटलं, आपण सॅम्पल घेतलं, अ रॅन्डम सॅम्पल ऑफ 6 सच बल्ब्स गेव द फॉलोइंग व्हॅल्यूज. 6 बल्ब घेतले आपण सॅम्पल म्हणून आणि त्यांचं रिअल फॅक्ट मध्ये डेटा कलेक्ट केला, ते म्हणजे तुमचं सॅम्पल आहे. द लाईफ बल्ब्स इन मंथ इज गिवन. पहिल्या बल्बचं लाईफ ऍक्च्युअल मध्ये बघितलं आपण 24 आहे, 24 मंथ्स. फॉर सेकंड बल्ब life is 26 months. For third bulb, life is actually 30 months. For fourth bulb, actual life is 20 months. For fifth bulb, the actual life is 20 months. And for last bulb, the actual life is 18 months. म्हणजे ऍक्च्युअली आपण 6 बल्ब सॅम्पल म्हणून निवडले आणि यांचं लाईफ ऍक्च्युअली काउंट केलं, ते म्हणजे आपलं सॅम्पल आहे. Sample size is only 6. Okay, but hypothetically or population म्हणून या ठिकाणी मीन आहे 25 months. कारण काय म्हटलंय, each bulb have bulbs have a mean life of 25 months. म्हणजे मीन लाईफ साधारणतः किंवा अर्थमेटिक मीन किती आहे यांचं 25 मंथ्स आहे. परंतु या ठिकाणी ऍक्च्युअली मध्ये 6 आपण सॅम्पल निवडले आणि त्यांचं ऍक्च्युअल लाईफ चेक केलं, मेजर केलं, तर आपल्याला असं लक्षात आलं 24, 26, 30, 20, 20 and 18 months. तुम्हाला पुढे प्रश्न विचारल्यावर आधारित, कॅन यू रिगार्ड द प्रोड्युसर्स क्लेम टू बी व्हॅल्यूड ॲट 1% लेवल ऑफ सिग्निफिकन्स. लेवल ऑफ सिग्निफिकन्स 1 ला 1% ला जे काही प्रोड्युसरनं क्लेम केला, कोणता क्लेम केला, की आमची कंपनीचा बल्ब हा 25 महिने चालेल साधारणतः, हा केलेला क्लेम व्हॅलिड आहे का, त्यात काही असोसिएशन आहे का, इज देयर एनी असोसिएशन, इज देयर एनी सिग्निफिकन्स डिफरन्स, इज देयर एनी रिलेशन, असं विचारलेलं आहे. आणि म्हटलं पुढे, गिवन दॅट द टेबल व्हॅल्यू फॉर डिग्रीज ऑफ फ्रीडम बी इज इक्वल टू n-1. n-1 या ठिकाणी तुम्हाला टेबल रोज आणि कॉलम्स मध्ये नाहीये, त्यामुळं जे काही डिग्रीज ऑफ फ्रीडम या ठिकाणी असेल ते म्हणजे व्हॉट एव्हर n इज देअर, फ्रॉम दॅट n you have to deduct 1, then you will get the degrees of freedom. So sample size is 6 minus 1, so degrees of freedom will be here 5. The level is 1%, means t's value, table value of t is equal to at 0.01 level, म्हणजे 1% level is 4.032. It is a table value and it is compared with the calculated value of t test, and then automatically we are able to conclude, we are able to interpret our hypothesis is accepted or rejected, and then we are going to take the proper meaning or interpretation of your result also. Okay, this is the problem. Now go to the solution. One minute. Okay. Okay. Is it visible the screen, please?
Yes, sir.
Okay.
Yes, sir.
Yes. Now solution same like chi-square test. First of all, you have to put the null hypothesis. And a hypothesis. We are here testing of hypothesis. And in testing of hypothesis, the first step is that firstly formulate the hypothesis. Firstly put the hypothesis. Then apply the formula. Then get the calculation. And lastly compare calculated value of whatever test is there with the table value. And on the basis of that, you have to conclude your whatever result is there means accepted or rejected, or whatever interpretation is or inference is there. So firstly here again null hypothesis we need to state, and again null hypothesis same form मध्ये करायचं बघा आता. Let us take the null hypothesis that there is no significant difference in the mean life of bulbs in the sample and that of the population. काय म्हटलं या ठिकाणी सॅम्पल मीन लाईफ आणि पॉप्युलेशनचं मीन लाईफ ऑफ द बल्ब सारखं आहे, त्याच्यात फरक नाही. देर इज नो सिग्निफिकन्स डिफरन्स. लक्षामध्ये देर इज नो सिग्निफिकंट डिफरन्स इन द मीन लाईफ ऑफ द बल्ब्स इन द सॅम्पल अँड दॅट ऑफ द पॉप्युलेशन. दुसऱ्या सेंटेन्स मध्ये आपण असं स्टेट करू शकता, there is no significant difference between the mean of sample and population. Sample चा mean आणि population चा mean यात काही फरक नाही असं म्हणणे म्हणजेच null hypothesis स्टेट करणे. Now apply t-test. It is one sample test, so apply the formula of one sample test. So t is equal to x bar minus mu. X bar means the mean of the sample, mu means mean of the population, into root n means size of the sample, s means divided by s means standard deviation. So one by one, where x bar is the mean of the sample, mu is the actual or hypothetical mean of the population. की जे कंपनीने दावा केला ना आमचा बल्ब हा 25 महिने साधारणतः चालेल, that is the hypothetical or population mean. Then n the size of sample means किती सॅम्पल आहे आपल्या बल्ब, किती बल्ब निवडले आपण 6 बल्ब निवडले 6, त्यामुळे सॅम्पल साइज इज 6. देन s स्टँड फॉर स्टँडर्ड डेविएशन ऑफ द सॅम्पल. तर एक सिंगल आहे या ठिकाणी, कंबाईन नाही, सिंगल सॅम्पल असल्यामुळं. तर या ठिकाणी लक्षात घ्या, मी मागे सुद्धा सांगितलं होतं स्टँडर्ड डेविएशनचा फॉर्म्युला किंवा स्टँडर्ड डेविएशन म्हणजे काय, की आपण काय करतो, whatever deviations we are going to take from all of, I mean from actual mean. Actual mean means what? X bar. X bar is the actual mean of the sample. We are going to deduct actual mean of the sample from all observations of sample. Then whatever value we will get, that will be squared. त्याचं काय करायचं, स्क्वेअर करायचं. स्क्वेअर केल्यानंतर सम करायचं. 6 व्हॅल्यू असेल तर सगळ्यांची बेरीज करा. डिवायडेड बाय n असतं जनरली स्टँडर्ड डेव्हिएशनच्या फॉर्म्युल्यामध्ये काय असते, डिवायडेड बाय n असतं. बट हियर इन केस ऑफ टेस्टिंग ऑफ हायपोथेसिस ऑलवेज स्टँडर्ड डेव्हिएशन इज कॅल्क्युलेटेड बाय डिवायडिंग द डिग्रीज ऑफ फ्रीडम, and here degrees of freedom is n-1. So not n only, so n-1 in case of only the testing of hypothesis while calculating the standard deviation. Okay, now go to the calculation of the mean, that is the x bar, the standard deviation of sample means yes, and lastly t value, which will comparable with the table value of t. So here firstly put the samples in x 24, 26, 30, 20, 20 and 80. Means the values of sample, whatever observations are there, only the values of sample and nothing else. Okay, Then x minus x bar means whatever actual mean is there. आपल्याला काढायचं actual mean. आपण काढू आता या ठिकाणी बघा. X bar means the sum of all divided by all. All means n. या 6 आकड्यांची बेरीज केली आपण 24 plus 26 plus 30 plus 20 plus 20 plus 18 is equal to sum of x 138. 138 is here. Divided by n. How many numbers are there? 1, 2, 3, 4, 5, 6. Sample size n 6. So the mean of the sample is 23. Then go for standard deviation. And in case of standard deviation, under standard formula, whatever x minus x bar is there, we have to take into the square. So x minus x bar जो आपको आ गया है उसका आप क्या करना है नेक्स्ट स्टेप में स्क्वायर करना है. ये x minus x bar है. x minus x bar whole square is equal to आता या ठिकाणी 24 minus 23 is equal to 1, 26 minus 23 is equal to 3, 30 minus 23 is equal to 7, 20 minus 23 is equal to minus 3, again 20 minus 23 is equal to minus 3, 18 minus 23 is equal to minus 5. Then take the square. The square of 1 is 1, the square of 3 is 9, the square of 7 is 49, the square of minus 3 is 9, the square of minus 3 is again 9, the square of minus 5 is 25. Now take the sum of these squared deviations from actual mean, and the sum is 102. So in case of standard deviation formula, under root sum of x minus x bar whole square is 102 divided by n minus 1. N is 6 minus 1 is equal to 5. So 102 divided by 5 is equal to 20.4. Now take the square root of the 20.4. The square root is 4.517. Okay, this is the standard deviation. Now go to the formula of the t-test. Now calculate t value is equal to mean of sample minus mean of population into under root sample size divided by divided by the standard deviation of the sample just calculated now. Now substitute the values. The mean of the sample is this one 23 minus mean of the population. In your problem given, the company claims the mean life of the bulb is 25 months. This is the mean of the population, hypothetical mean 25 into under root size of sample n is 6 divided by yes. Yes means just calculated the standard deviation value 4.517. Now go to the calculation. Take the just difference instead of sign. 23 minus 25 is equal to 2. Then the square root of the 6 is 2.449. The multiplication of 2 into 2.449 is equal to 4.899 divided by 4.517. Then take the division. Answer is 1.084. Now this is the calculated value of t-test, and it will be compared with the table value of t, and then we are going to decide our null hypothesis is accepted or rejected, and then we are going to interpret the meaning of the test, meaning of the result, and we are going to inference the conclusion. Now step second. The table value of the t for the degrees of freedom 5 is at 0.01 level or 0.1% level of significance is already given in the problem. That is a 4.032. It is a calculated value 1.084 and 4.032 is the table value of t. Now compare the calculated value with the table value and then decide hypothesis is accepted or rejected. Okay. the calculated value of t, that is 1.084, is less than the table value of t, that is 4.032 at 1% level of significance and 5 degrees of freedom. Hence, the null hypothesis is accepted. जेव्हा calculated value कमी असते तेव्हा null hypothesis accept होतं, otherwise reject होतं. It means that there is no significant difference in the mean life of bulbs in the sample and that of the population. Sampleचं mean life आणि populationचं mean life यामध्ये लक्षात घेण्यासारखा फरक नाही. There is no significant difference. आपण जे स्टेट केलं होतं हायपोथेसिस ते ऍक्सेप्ट झालं, म्हणजे मिनिंग लिहिताना ऍज इट इज खाली लिहायचं. म्हणजेच काय, प्रश्न विचारला काय आपल्याला बघा, द प्रोड्युसर्स क्लेम इज व्हॅल्यूड ऍट 1% लेवल ऑफ सिग्निफिकन्स. प्रोड्युसरनं क्लेम काय केला होता, आमच्या बल्बचं साधारणतः 25 महिने हे लाईफ असतं. हा क्लेम केलेला बरोबर आहे. का बरोबर आहे? कारण सॅम्पल मीन आणि पॉप्युलेशन मीन याच्यात फरक नाही. These two are similar अशा अर्थानं उत्तर आलेलं आहे. There is no significant difference in between the mean of the sample and mean of the population. आणि म्हणून प्रोड्युसरने केलेला क्लेम हा या ठिकाणी काय आहे, व्हॅलिड आहे. ओके, ओव्हर.
त्याला मेंढूळ आलं, मेंढूळ. सर, हॅलो हॅलो.
हॅलो, आवाज येतो का माझा आता?
Yes, sir. You are audible. Yes, sir. You are audible, sir.
इको होतोय सर पण.
ओके ओके. आर्लियर
सर कोणतं तरी एक माईक बंद करायचं, नाही तर त्याचा आवाज बंद करायचा
मी
लॅपटॉपचा.
मी एक बंद करतोय. अगोदर मला रिस्पॉन्स येत गेला म्हणून चालू केला आता. ठीक. हॅलो,
हा सर.
एक 1 मिनिट थोडं वेट करा. एक प्रॉब्लेम डिस्प्ले करतो. ओके. हॅलो, थोडा प्रॉब्लेम बघा तुम्ही. मी 2 मिनिटात
हा, सर.
करतो. ओके. Okay, sorry for disturbance. Now I am audible, please. Am I audible?
Yes, sir. You are audible, sir.
Okay. Technical problem is there. This is the problem number 2. Okay. The life of time. The life of time of electric bulbs for a random sample of 10 bulbs from a large consignment give the following data. आपल्याकडे खूप मोठं बल्बचं कन्साइनमेंट आहे, पार्सल आहे. त्यामध्ये आपण 10 बल्ब रॅन्डमली सॅम्पल केले. ओके. Can we accept the hypothesis that the average life of bulb is 4000 hours? आपल्याला म्हटलेलं आहे, तुम्ही हे ऍक्सेप्ट कराल का हायपोथेसिस की साधारणतः त्याचं ऍव्हरेज लाईफ हे 4000 आवर्स आहे. मागचा प्रॉब्लेम मंथ्स मध्ये होता, हा प्रॉब्लेम आवर्स मध्ये आहे. एवढंच फरक. तर साधारणतः 4000 तास सरासरी लाईफ आहे. हे तुम्हाला पटतं का? तुम्हाला ऍक्सेप्ट होईल का? याच्यामध्ये म्हणजे हे पॉप्युलेशन मीन आहे. कुठलं मीन आहे? 4000 hours is a population mean. आणि मग या 10 सॅम्पलचं काय ऍक्च्युअल लाईफ आहे हे पुढे दिलेलं आहे बघा. Given that the table value of for degrees of freedom n-1 that is 10-19 at 5% level, मागचं 1% लेवल होतं, आता या ठिकाणी टी काय आहे 5% लेवल आहे, डिग्रीज ऑफ फ्रीडम 9 आहे, टी इज इक्वल टू at 0.05 level is equal to 2.262. ही टेबल व्हॅल्यू आहे आणि तुम्हाला डेटा खाली दिलेला आहे. काय दिलेला आहे, किती बल्ब तुम्ही सॅम्पल निवडले 10, म्हणून या ठिकाणी काय म्हटलेलं आहे 10 निवडले म्हणल्यामुळं तुम्हाला या ठिकाणी ऍटम्स म्हटलेलं आहे. पहिला फर्स्ट बल्ब, सेकंड बल्ब, थर्ड बल्ब, फोर्थ बल्ब, फिफ्थ बल्ब, सिक्स्थ बल्ब, सेवंथ बल्ब, एट्थ वन, ninth one and 10th one sample of the bulb. या 10 बल्ब तुम्ही सॅम्पल निवडलात आणि त्यांचं लाईफ तुम्ही ऍक्च्युअली मेजर केलात आणि हे मेजर केलेलं लाईफ बघा, लाईफ टाइम इन 1000 hours means whatever the figures are here in the table is are stating in thousands. So 4.2 thousand, 4.6 thousand, 3.9 thousand, 4.1 thousand, 5.2 thousand, 3.8 thousand, 3.9 thousand, 4.3 thousand, 4.4 thousand, 5.6 thousand. These are the actual reading of the sample. Means actual life of these 10 bulbs. And our hypothetical life is given 4000 hours. So तुम्हाला काय विचारलं, can we accept the hypothesis that our average life of time of bulbs is 4000 hours, means population mean तुमचं 4000 आहे आणि actual mean तुम्हाला काढायचं आहे आणि नंतर compare करायचं आहे. Now go to the solution. Firstly, put the hypothesis. Let us take the null hypothesis that there is no significant difference in the sample mean and the population mean or in the mean of sample and population. असं म्हणत तरी चालते. म्हणजेच पॉप्युलेशनचं मीन 4000 आणि सॅम्पलचं मीन जे तुम्ही काढाल ते यात काही फरक नाही, म्हणजे नल हायपोथेसिस आहे. नाउ अप्लाय टी टेस्ट. अगेन इट इज वन सॅम्पल टेस्ट, सो अप्लाय द फर्स्ट फॉर्म्युला. T is equal to mean of the sample minus mean of the population mu into root under root n means size of the sample 10 divided by s means the standard deviation of the sample. So here the mean of the sample we need to calculate the hypothetical mean of the population, that is mu is equal to already given in the problem 4000 hours. A problem दिला बघा सुरुवातीला at the beginning. Okay, now go next. The sample size is 10. एकूण sample size किती आहे 10 आहे, 10. Now go okay, now go. ठीक है. Then standard deviation is equal to this formula. Deviations taken from actual mean then square divided by n minus 1 is the solution of standard deviation. Now put the values in formula and get the answer. Calculation of the mean x bar, the calculation of standard deviation, that is yes, and t value. So here firstly put the values as x. These all values are in 1000 hours. So 4.2 thousand, 4.6 thousand, 3.9 thousand, 4.1 thousand, 4.2 thousand, 3.8 thousand, 3.9 thousand, 4.3 and 4.4 and 5.6 thousand. Total 44,000. Total 44,000. Now calculate the arithmetic mean. And arithmetic mean is equal to sum of all divided by all. Sum of all is 44 divided by all observations, n, that is 10, is equal to 4.4 thousand hours, because these are all figures in 1000 hours. So actual mean of the sample is 4.4 thousand hours. Now standard deviation. Go for standard deviation. In case of standard deviation, we need to take into account the deviations from actual mean, then square and then divide by degrees of freedom and then take the root. So firstly, these are x values minus x bar. X bar means this is a 4.4. 4.4. So take into account 4.2 minus 4.4 is equal to minus 0.2. 4.6 minus 4.4 is equal to plus 0.2. 3.9 minus 4.4 is equal to minus 0.5. 4.1 minus 4.4 is equal to 0.3. 5.2 minus 4.4, 4.4, then is equal to is equal to 5.2 minus 4.4 is equal to 0.8. 3.8 minus 4.4 is equal to minus 0.6. 3.9 minus 4.4 is equal to 0. Minus 0.5. 4.3 minus 4.4 is equal to 0 minus 0.1. 4.4 minus 4.4 is equal to 0. 5.6 minus 4.4 is equal to 1.2. Now take this take the square of such deviations. X minus x bar whole square means take the square. Minus 0.2 into minus 0.2 is equal to 0.04. 0.04 square, then again 0.04 square, 5 is at 0.25, 3 is 0.09, 8 is 0.64, 6 is 0.36, 5 is 0.25, 1 is 0.01, and 0 into 0 is equal to 0. 1.2 into 1.2 is equal to 1.44. Now take the sum of these square deviations, and the sum of the square deviation is 3.12. 3.12. Now put the values in formula of standard deviation. Standard deviation is equal to under root x minus x bar whole square sum. X minus x bar whole square sum means 3.12. 3.12. Then divided by n minus 1. N is a 10. Minus 1 is equal to 9. So 3.12 divided by 9, you will get the calculation of division 0.3467. 3467. Now take the square root of this one. It is 0.589. This is the standard deviation. This is the standard deviation. Okay. Now go to calculate the t value. So t value is equal to mean of the sample minus mean of the population into root size of sample divided by standard deviation. So sample mean just calculated 4.4 thousand minus population mean already given 4,000 hours. So minus 4 into n means 10 under root 10 divided by s means standard deviation just calculated 0.589 is equal to, put the value, get the answers. 4.4 minus 4 is equal to 0.4 into the square root of the square root of the 10 is equal to 3.1622 divided by, as it is, 0.589. Then take the division of these last two, and your answer will be 2.1476. Now, this is the calculated value of t, will be compared to the table value, and then we are going to decide the hypothesis is accepted or rejected. So the table value of t for degrees of freedom v, for degrees of freedom v is equal to n minus 1, that is 10 minus 1 is equal to 9, and level of significance is already given, 0.05 means 5% level is equal to, already given in the problem, 2.262. Now compare the calculated value of t with the table value. The calculated the calculated value of t is 2.1476, is less than the table value of t, that is 2.262 at 5% level of significance. Hence, the null hypothesis is again accepted here. It means that there is no significant difference in the sample mean and the population mean, or in difference in mean of the sample and that of the population. We can accept the hypothesis that the average life of bulb is 4000 hours. तुम्ही मला विचारला प्रश्न, साधारणतः 4000 तास बल्ब चालतो, हे हायपोथेसिस स्वीकाराल का? स्वीकारणारच. कारण काय की पॉप्युलेशन मीन आणि सॅम्पल मीन यात फरक नाही. There is no significant difference in between the mean of the sample and mean of the population. That's why whatever population mean is there, I mean hypothetical mean is there, that we are accepted here. Okay. काय अडचण असेल ते विचारा.
Hello, sir.
बोला.
See, sir, in this column which you have flashed here, no, sir, we calculate x minus x bar. Okay. And after that, we take their squares. So last
Yes.
value of that x minus x bar, that is 1.2, sir. Okay. So can we
Yes.
can't we directly square it, sir? Like, if there is a need to once again to calculate square of minus 0.2, square of minus 0.5, or directly we can calculate the square of that final answer. That 1.2 square is 144, like 1.44, like that.
Means instead of second column, you want to directly use third column.
No,
Yes,
no.
1 minute.
Can we do
No,
that?
no, 1
sir?
minute. Yes, yeah, why not? Why not? 1 minute, sir. After calculation of arithmetic mean, okay, we are taking the deviations only for standard deviation. And in case of standard deviation, we require square of the deviations and sum. So whatever difference is there in between x and x bar, you can directly take the square also. Okay.
Yes, sir. That only I wanted to ask, because
No,
to
these
the
are the
point
just
of-
steps, sir. These are the
Yes,
steps. कैसा है?
No, no, no. That only I want to ask you, sir, that are these steps necessary for the coursework exam to write there, or directly we can find it out?
Okay,
Like-
okay. Here we need only the last column, because first column is useful for calculation of arithmetic mean. After mean, we require the last column. I mean x minus x bar whole square sum we require. Okay.
Yes,
If you
sir.
are
Yes,
taking
sir.
directly square, it is okay, no doubt.
Yes,
You are right. Okay.
yes,
But
sir.
I mean, while taking square, if any missing is there while taking square, that will be very difficult to find your answer is correct or not till the result. That's why you put firstly the deviations and then the square it. It is better. Okay.
Yes, sir. Yes, sir. Understood.
Okay.
sir. Thank
Again,
you,
anybody? This is the one sample test. Now we are going to start the two sample tests. Okay. ही वन सॅम्पल टेस्ट होती. आता आपल्याला टू सॅम्पल घ्यायचे. चालेल, काय अडचण असेल तर विचारा. फार सोपं आहे. इझी वे मध्ये करायचं, कन्फ्युजन नाही करायचं. क्लॅरिटी इज व्हेरी इम्पॉर्टंट इन कॅल्क्युलेशन. इफ वी आर कन्फ्युज, then we are unable to go to the proper solution. Okay. Let me start the next method, next type of t-test.
Yes, sir.
Okay, okay. Problem number 3. Okay. Is it visible the screen?
Yes, sir. Yes, sir.
Okay. Now problem number 3 on t-test. Problem number 3. Two types of drugs were used on 5 and 7 patients for reducing their weight. आपण काय केलं, वेट कमी करण्यासाठी 5 पेशंटचा एक ग्रुप आहे आणि 7 पेशंटचा एक ग्रुप आहे. त्यांच्यावर 2 टाईपचे ड्रग यूज केले. म्हणजे 5 लोकांवर वेगळा ड्रग आणि 7 लोकांवर वेगळा ड्रग. तर काय झालं, रिझल्ट काय म्हणतो? ड्रग ए वॉज इम्पोर्टेड, हा आयात केलेला होता, आणि ड्रग बी इंडिजिनियस, तो भारतीय होता. The decrease in the weight in kg after using the drugs for 6 months was as follows. या 5 लोकांना आणि 7 लोकांना, 5 लोकांना ड्रग ए दिला आणि 7 लोकांना ड्रग बी दिला, आणि 6 महिन्यानंतर त्यांचं वेट केलं. ते वेट किती कमी झालं, कारण एक्सक्लुझिव्हली वी आर गोइंग टू ट्रीट द फॉर रिड्यूसिंग देअर वेट. So after 6 months, when we observe the data, in case of sample of 5 patient, drug A, and in case of sample 2, sample size, second sample size of 7 people, drug B, आणि 6 महिन्यानंतर आपल्याला त्यांचे फिगर्स आले, एवढे किती वजन कमी झालं. So in case of first sample means the patients using drug A, the weight reduced during 6 months is 10 kilograms, 12 kilograms, 13 kilograms, 11 kilograms, and 14 kilograms. Then the reduction in weight of the patients who are using drug B. Drug B वापरण्याच्या ठिकाणी बघितलं तर ड्रग बी वापरणे वजन कमी झालं बघा. पहिल्याचं 8 kg, सेकंड 9 kg, थर्ड वन 12 kg, fourth one 14 kg, fifth one 15 kg, sixth one 10 kg, and last one 9 kg. The weight is reduced during 6 months due to apply of drug B. Now next question for you is that, is there a significant difference in the efficacy of the two drugs? या दोन ड्रगच्या प्रभावीपणामध्ये काही फरक आहे का? If not, जर फरक नाही, दोन्ही सारखे आले सुदैवाने, म्हणजे नल हायपोथेसिस ऍक्सेप्ट झालं. इफ नॉट, जर फरक नाही, which drug should you buy? या दोन ड्रग पैकी कुठलं ड्रग तुम्ही खरेदी करायला पाहिजे? की जेव्हा डिग्रीज ऑफ फ्रीडम या ठिकाणी काय आलेलं आहे, 10 आलेलं आहे. 10 म्हणजे काय, या N1 मधून 1 वजा करा आणि या N2 मधून 1 वजा करा. 5 वजा 1 4 आणि 7 वजा 1 6, 4 आणि 6 10. Degree of freedom is here 10. Level of significance is 5%, that is 0.05, and the table value is given at this degrees of freedom, and level of significance is 2.228 and nothing else. Now go to the solution. First of all, put the hypothesis, or particularly null hypothesis. Let us take the hypothesis, null hypothesis, that there is no significant difference in the efficacy of two drugs. या दोन ड्रग्सच्या, आता हे 2 सॅम्पल टेस्ट आहे, यामध्ये पॉप्युलेशन मीन राहणार नाही. यामध्ये काय, दोन्ही सॅम्पलचं मीन आहे, त्यामुळे 2 सॅम्पल्स आहेत. आणि काय म्हणायचं या ठिकाणी, या दोन्ही ड्रग्सच्या इफिकेसी मध्ये, परिणामकारकतेमध्ये लक्षात घेण्यासारखा फरक नाही, म्हणजे त्यांच्यात काही फरक नाही. these two are equivalent, these two are independent, these two are any अशा अर्थाने म्हटलेले आहे. there is no significant difference in the efficacy of the two drugs. या दोन्ही ड्रग्सच्या इफिकेसी मध्ये काही फरक नाही, किंवा इन द इफिकेसी ऑफ drug 1 with the efficacy of the drug 2 असंही म्हटलं चालतं, किंवा असं डायरेक्टली म्हटलं चालतं. नाउ अप्लाय टी टेस्ट. आता या ठिकाणी टू सॅम्पल टेस्ट असल्यामुळं द फॉर्म्युला ऑफ टू सॅम्पल टेस्ट इज ऍप्लिकेबल हियर. सो टी इज इक्वल टू x1 bar means mean of the first sample minus x2 bar means mean of the second sample, means instead of population mean, here the mean of second sample will be there, divided by again the combined standard deviation of two samples. मागच्या मध्ये एकच सॅम्पल होतं, परंतु या ठिकाणी दोन सॅम्पल वेगळे आहेत, 5 आणि 7, त्यामुळे यांचं कंबाइंड स्टँडर्ड डेव्हिएशन असेल. into under root n1 means size of first sample multiplied by n2 means size of second sample divided by n1 size of first sample plus size of second sample. म्हणजे दोघांची बेरीज घ्यायची, 7 5 आणि 7 12, त्याच्यामध्ये दोघांचा गुणाकार भागिले दोघांची बेरीज. नाउ गो टू द ओके. येस बघा या ठिकाणी येस स्टँड फॉर द कंबाइंड स्टँडर्ड डेव्हिएशन ऑफ टू सॅम्पल्स. दोन्ही सॅम्पल्स हे कंबाइंड स्टँडर्ड डेव्हिएशन आहे. इज इक्वल टू, येस इज इक्वल टू व्हॉट, येस इज इक्वल टू अंडर रूट फर्स्टली deviations taken from actual mean for sample 1. So sum of the x1 minus x1 bar whole square, in addition to that for second sample again plus sum of x2 minus x2 bar whole square. दोन्ही बेरजा घ्यायच्या या ठिकाणी. हे पहिल्या सॅम्पलचं घ्यायचं, हे दुसऱ्या सॅम्पलचं घ्यायचं, डिवायडेड बाय डिग्रीज ऑफ फ्रीडम. डिग्रीज ऑफ फ्रीडम मीन्स एकच सॅम्पल असतं तर n-1, परंतु या ठिकाणी दोन सॅम्पल आहेत. दोन सॅम्पल म्हणजे n1 पहिल्या सॅम्पलची साईज, n2 म्हणजे दुसऱ्या सॅम्पलची साईज, मायनस दोन सॅम्पल मुळे -2. नाउ गो टू द कॅल्क्युलेशन. कॅल्क्युलेशन ऑफ टू मीन्स x1 बार अँड x2 बार. मागच्या प्रॉब्लेम मध्ये एकच मीन काढायचं होतं आणि पॉप्युलेशन मीन दिलेलं होतं, परंतु या ठिकाणी टू सॅम्पल असल्यामुळं आपल्याला दोन्ही सॅम्पलचं मीन काढावं लागतं. म्हणून x1 बार अँड x2 बार अँड द स्टँडर्ड डेव्हिएशन काढायचं आपल्याला, ते मात्र कंबाईंड आहे सॅम्पलचं, आणि नंतर टी व्हॅल्यू काढायची. ओके. आता दोन सॅम्पल असल्यामुळे मीन बदललं, लक्षात आलं का? गडबड करायची नाही. एका सॅम्पल स्वतंत्र घ्यायचं, दुसऱ्या सॅम्पल स्वतंत्र घ्यायचं, धावपळ करायची नाही. दोघांचं स्वतंत्र काढायचं अँड पुट द व्हॅल्यूज प्रॉपरली. सो हियर x1 फर्स्ट सॅम्पल 5 आकडे आहेत त्याच्यामध्ये, त्यामध्ये 10 प्लस 12 प्लस 13 प्लस 11 प्लस 14. द ऍडिशन ऑफ these all is equal to 60. So 60 divided by n1 किती आहे, साइज किती आहे, 5. 60 divided by 5 is equal to 12. So the mean of the first sample is 12. Sum of all divided by all, your answer is 12. Now go for the second sample. Second sample x2. The addition of 8, 9, 12, 14, 15, 10, 9 is equal to 77. Total number of observations are 1, 2, 3, 4, 5, 6, 7. So 77. Sum of x2 is 77 divided by n2 means 7. So 11 will be the mean of second sample. Now take the deviations from actual mean in case of first sample and in case of second sample one by one. So firstly x1 minus x1 bar. This one is the x1 bar. What? This one is the x1 bar. So here x1 minus x1 bar is equal to 10 minus 12 is equal to minus 2, 12 minus 12 is equal to 0, 13 minus 12 is equal to 1, 11 minus 12 is equal to minus 1, 14 minus 12 is equal to 2. Then take the square. Minus 2 square 4, 0, 1, 1 and 4. The sum of these all squared is equal to 10. So it is called as sum of x1 minus x1 bar whole square is equal to Sarvanchi is 10. Now go for second sample. In case of second sample, x2 minus x2 bar means 8 minus 11 minus 3, 9 minus 11 minus 2, 12 minus 11 1, 14 minus 11 3, 15 minus 11 4, 10 minus 11 minus 1 and 9 minus 11 minus 2. Now take the square. Square is 9, 4, 1, 9, 16, 1 and 4. Now take the sum of the square. It is 44. Okay. It is applicable for standard deviation. Okay. Now go to calculate the standard deviation. The combined standard deviation is equal to under root. Firstly, sum of the x minus x1 minus x1 bar whole square means 10. Means take the 10. Secondly, sum of another means 44 divided by n1 means 5, n2 means 7, minus 2 minus 2. So 10 plus 44 divided by 5 plus 7 minus 2 is equal to 10. 54 upon 10 is 5.4 into root. The square root will come 2.32238. Okay, this is the standard deviation. After standard deviation, go for the t value. T value is equal to what? X bar minus x1 bar minus x2 bar divided by standard deviation into under root n1 multiplied by n2 divided by n1 plus n2. So this mean of first sample 12, mean of the second sample 11 divided by standard deviation 2.3238 in root n1 into n1 into n2 means 5 into 7 divided by addition of these two 5 plus 7. So next 12 minus 11 1 divided by 2.3238 into 5 7 35 divided by 5 plus 7 12. Next 1 upon the division of 35 divided by 12 is equal to 2.9167. Then the square root of this one will come 1.7078 into 1 is equal to same divided by 2.3238 is equal to 0.7349 is the value of t, calculated value of t. Now the table value of t for degrees of freedom n1 plus n2 minus 2, 5 plus 7 minus 2 is equal to 10 at 0.05 level is already given in the problem 2.228. Now compare your calculated value of t with the table value and then decide the hypothesis is accepted or rejected. The calculated value of t, that is 0.7349, is less than the table value of t, that is 2.228 at 5% level of significance. Hence, the null hypothesis is accepted. It means that there is no significant difference in the efficacy of two drugs. Null hypothesis accept झालं म्हणजे दोघांच्या परिणामकारकतेमध्ये फरक नाही. Means ए ड्रग ऍज वेल ऍज बी ड्रग दोन्हीही सारखे आहेत, त्यांच्यात फरक नाही असा आपण निष्कर्ष काढला. मग तुम्हाला त्यांनी प्रश्न विचारला, ड्रग ए हे इम्पोर्टेड आहे आणि ड्रग बी हे इंडिजिनियस आहे, तर मग दोन्हीपैकी कोणतं ड्रग खरेदी कराल? कोणतं कराल? कोणतं कराल? भारतीय कराल का विदेशी कराल? जर दोन्ही सारखे असतील तर कोणतं कराल
Indian.
सांगा.
दोन्ही दोन्ही
indigenous one.
आन्सर
Indigenous
चूक
one.
आहे, लक्षात घ्या. If there is no significant difference in between the drug A which is imported and drug B which is indigenous, दोन्ही जर सारखे असतील तर तुम्हाला प्रश्न विचारला ऍज अ ह्युमन बिंग ऑफ द इंडियन सिटीजन, विच वन शुड यू बाय? तुम कौन सा खरीदी करना चाहेंगे?
Indian
So
drug.
it is better to say if these two are same, it means it is better to buy from India or indigenous, that is B. Since B drug is indigenous and there is no any significant difference in the efficacy of imported as well as indigenous drug, so we should buy indigenous drug, that is B. B खरेदी करायला पाहिजे. लक्षामध्ये बाहेरची खरेदी केल्यामुळे बरं वाटतं म्हणून असं नाही करायचं. लक्षामध्ये जर सारखं असेल तर, चांगलं असेल तर काय हरकत नाही. ओके ओके, थँक्यू. काय अडचण असेल ते विचारा.
नाही सर.
नाउ गो फॉर प्रॉब्लेम नंबर 4. Okay, problem number 4. It is on two sample test also. For a random sample of 10 persons, 10 लोकांचे एक सॅम्पल आहे. फेड ऑन डाएट ए, यांना डाएट ए दिला. द इन्क्रीज वेट इन किलोग्राम्स इन अ सर्टेन पिरियड वेअर, एका विशिष्ट कालावधीमध्ये त्यांचं वजन किती वाढलं त्याचे आकडे आहेत. पहिल्याचं 10 किलो वाढलं, दुसऱ्याचं 6 किलो, तिसऱ्याचं 16 किलो, चौथ्याचं 17 किलो, 13 kg, 12 kg, 8 kg, 14 kg, 15 kg, and last one's 9 kg. This is the weight increased during a certain period, maybe 1 month, 2 months, 3 months, like that. And the diet A was given to these 10 persons. या 10 लोकांना डाएट कुठला दिलेला आहे? डाएट ए दिला आणि वजन मोजलं, एवढं वजन त्यांचं या काळात वाढलं. This is the sample 1. For another random sample of 12 persons fed on diet B, दुसरे 12 लोक आहेत, त्यांना डाएट बी दिला. द इन्क्रीज वेट इन किलोग्राम्स इन अ सर्टेन पिरियड, त्याच कालावधीमध्ये वजन वाढलेलं किलोग्राम्स मध्ये मोजलं तर असं झालं. 7 kg, 13 kg, 22 kg, likewise up to 17 kg of last one. टेस्ट, तुम्हाला टेस्ट करा म्हटलं, काय करा? टेस्ट whether the diets A and B differ. डाएट ए आणि बी वेगळे आहेत का? आपण नाहीत असं म्हणतो अगोदर, त्याला म्हणायचं नल हायपोथेसिस. सेम आताच्या सारखंच. डिफर सिग्निफिकंटली, लक्षात घेण्यासारखा दोघात फरक आहे का? ऍज रिगार्ड्स देअर इफेक्ट, त्यांचा परिणामकारकतेमध्ये. ऑन द इन्क्रीज इन वेट, वजन वाढवण्यामध्ये, परिणाम करण्यामध्ये लक्षात घेण्यासारखा फरक आहे का? गिवन द फॉलोइंग. आता या ठिकाणी गिवन द फॉलोइंग मध्ये काय केलंय, तुम्हाला फक्त टेबल व्हॅल्यू दिलेले आहे. कुठल्या डिग्रीज ऑफ फ्रीडमला कुठली टेबल व्हॅल्यू आहे, बाकी काही नाही. सो यार डिग्रीज ऑफ फ्रीडम 19 असेल तर टेबल व्हॅल्यू 5% लेवलला एवढी आहे. डिग्रीज ऑफ फ्रीडम 20 असेल तर हे वापरा. डिग्रीज ऑफ फ्रीडम 21 असेल तर ही व्हॅल्यू वापरा. डिग्रीज ऑफ फ्रीडम 22 असेल तर ही वापरा. डिग्रीज ऑफ फ्रीडम 23 असेल तर ही वापरा. जे डिग्रीज ऑफ फ्रीडम तुम्हाला अप्लिकेबल आहे ही टेबल व्हॅल्यू वापरा असं म्हटलं, बाकी काही नाही. नाउ solution of the problem number 4. Again, put the null hypothesis same as earlier. Let us take the null hypothesis that there is no significant difference between diet A and diet B as regards their effect on the increase in weight. डाएट ए आणि डाएट बी मध्ये कुठलाही लक्षात घेण्यासारखा फरक नाही की ज्याच्यामुळे वजन लोकांचं वाढेल. फरक नाहीये. वाढलं नाही वाढलं फरक नाही, सारखे. Apply T test, particularly to sample test. So formula again is here. I don't want to speak again and again. Formula you know well. X1 bar means sample mean of the first sample. X2 bar means mean of the second sample. S means the combined standard deviation. N1 means the size of first sample. N2 means size of second sample. Okay. Again, standard deviation are similar, which is earlier studied. Okay. Firstly, take the deviation square. Deviations from actual mean for first sample. Then second divided by N1 plus N2. Degree of freedom is equal to -2. Now calculation of 2 means X1 bar, X2 bar. Then standard deviation, that is combined standard deviation, and lastly T value we need to calculate. So here again, X1 sample is of 10 size and X2 sample is of 12 size. Okay. So you take the means first, means the sum of these all divided by all is equal to 120 upon 10 is equal to 12. For second sample, the sum of all is 180 divided by all means how many items are there? 12. So 180 divided by 12 is equal to 15. So 12 is the mean of first sample and 15 is the mean of second sample. Now go for next step, that is the standard deviation. In case of standard deviation, square deviations are there in case of first sample, in case of second sample. So first we take the deviations and then square it and sum it. Okay. So 10 minus 12 is equal to -2, 6 minus 12 is equal to -6, 16 minus 12 is equal to 4, 17 minus 12 is equal to 5, 13 minus 12 is equal to 1, 12 minus 12 0, 8 minus 12 minus 4, 14 minus 12 is equal to +2, 15 minus 12 is equal to 3, 9 minus 12 is equal to -3. Now square 2 minus 2 square 4, 36, 16, 25, 1, 4, 16, 4, 9 and 9. The sum of the square deviation for in case of first sample is equal to 120. So put the 120. Okay. Sum of x minus x bar x1 bar square is equal to 120. Now go for second sample. In case of second sample, 7 minus 15 is equal to -8. 13 minus 15 is equal to -2. 22 minus 15 is equal to 7. 15 minus 15 is equal to 0. 12 minus 15 -3. 14 minus 15 -1. 18 minus 15 3. 8 minus 15 -7. 21 minus 15 is equal to +6. 23 minus 15 is equal to 8. And 10 minus 15 is equal to -5. 17 minus 15 is equal to 2. Now take the square of the such deviations. 64, 4, 49, 0, 9, 1, 9, 49, 36, 64, 25, 0, 4. Now take the sum of these all square deviations. Square deviations is equal to 314. You put here 314. For first one, 120. For second one, 314 divided by N1 is equal to 10 plus N2 is equal to 12 minus 2 is equal to 20. So 434 divided by 20 is equal to 21.7. Now the square root of the 21.7 is 4.6583 is the standard deviation. Now apply the t test. T test is equal to mean of the first sample minus mean of the second sample divided combined standard deviation into N1 10 into 12 divided by 10 plus 12. Then you will get first sample mean 12 minus second sample mean 15 divided by standard deviation 4.6583 into root 10 into 12 and 10 plus 12. Go for next. Difference is 3 divided by 4.6583 into the multiplication 120 divided by addition 22. Then 3 upon the division of 120 upon 22 will come 5 point under root 5.4545. Then take the square root. Square root is 2.3355 into 3 divided by same. The multiplication of these two is 7.0065 divided by 4.6583 is equal to 1.5040. This is the t value. Now, table value of t for degrees of freedom 10 plus 12 minus 2, that is 20, is equal to at 0.05 level 2.09, and calculated value is 1.50. So the calculated value of t 1.5040 is less than the table value of t 2.09 at 5% level of significance. Hence, the null hypothesis is accepted. It means that there is no significance difference between diet A and diet B as regards their effect on this increase in weight. दोन्ही डाएट मध्ये काही फरक नाहीये. दोन्ही डाएट मुळे वजन त्याच प्रमाणात वाढतं, वेगळ्या पद्धतीने काही होत नाही. म्हणून there is no any significant difference between diet A and diet B as regards their effect on the increase in weight is same. Okay. सांगा काय अडचण असेल तर.
नाही सर.
किती वेळ राहील आपल्याकडे?
अर्धा तास आहे सर अजून.
ठीक आहे, आपण काय करूयात मग, आणखी एक एफ टेस्ट असते. एफ टेस्ट म्हणजे व्हरायन्स टेस्ट लक्षामध्ये, आणि व्हरायंट टेस्ट बद्दल आपण काही घेऊ आहे का नाही?
हो सर.
म्हणजे व्हरायंट टेस्ट सुद्धा एक पॅरामेट्रिक टेस्ट आहे. आता ही मीन टेस्ट होती, अगोदरची चाय स्क्वेअर टेस्ट ही ऍट्रिब्यूट टेस्ट आहे. दोन ऍट्रिब्यूट आपण टेस्ट करता, आणि टी टेस्ट मध्ये मीन टेस्ट करता आपण, सॅम्पल मीन विथ पॉप्युलेशन मीन किंवा टू सॅम्पलचं डिफरंट मीन असेल तर आपण आता एफ टेस्ट मध्ये या ठिकाणी व्हरायंट टेस्ट केली जाते. तर मी शेअर करतो या ठिकाणी, दुसरीकडनं करतोय. एक मिनिट. स्क्रीन शेअर दुसरं करतोय मी. 1 मिनिट. ओके, दिसते का स्क्रीन? स्क्रीन दिसते का सर्वांना?
हो सर, दिसतंय सर.
Yes, sir.
आवाज एका साइडने आहे, स्क्रीन शेअरिंग वेगळ्या डिव्हाइसवरनं आहे. ओके, एफ टेस्ट थेरी पार्ट बघू आपण, नंतर प्रॉब्लेम घ्यायचा. Hypothesis testing, F test. The F test is named as named in honor of the great statistician R.A. Fisher. म्हणून याला F म्हणतात. The object of F test is to find out whether the two independent estimates of population variance differ significantly. म्हणजे दोन पॉप्युलेशनचे जे व्हरायन्स आहेत लक्षात घेण्यासारखे वेगळे असतील तर यासाठी आपण टेस्ट वापरत आहे. और whether the two samples may be regarded as drawn from the normal populations having the same variance. Variance means what? Square of the standard deviation. जे स्क्वेअर रूट आपण करता ते स्क्वेअर रूट आपण काढून टाकलं स्टँडर्ड डेव्हिएशनचं उत्तर येतं व्हरायन्स. A statistical test, F test uses and F statistics. जसं टी टेस्ट साठी टी टेबल आहे, चाय स्क्वेअर साठी त्याचा टेबल आहे, असं एफ स्टॅटिस्टिक या ठिकाणी आहे. टू कम्पेअर द टू व्हरायन्सेस S1 आणि S2. ओके, या ठिकाणी S1 आणि S2 या ठिकाणी काय म्हटलेलं आहे, स्टँडर्ड डेव्हिएशन वापरलं नाहीये तर व्हरायन्स म्हणून वापरलं. लक्षात घ्या. बाय त्याला स्क्वेअर करतात आपल्यानंतर बाय डिवायडिंग देम. दोन्ही जे व्हरायन्स तुम्ही काढता ते काढताना S1 आणि S2 या एकमेकाला डिव्हाइड करायचे आहे आणि उत्तर काढायचं असते. या ठिकाणी द रिझल्ट इज ऑलवेज पॉझिटिव्ह. या ठिकाणी रिझल्ट पॉझिटिव्ह असतो. त्याचं कारण असं आहे की मोठा मोठा व्हरायन्स हा बिगर किंवा लार्जर व्हरायन्स हा आपण वर लिहितो आणि स्मॉलर व्हरायन्स हा आपण डिव्हिजन मध्ये लिहितो. म्हणून येणारं आन्सर हे आपलं जनरली प्लस मध्ये येते अशा अर्थाने म्हटलंय. द रिझल्ट इज ऑलवेज अ पॉझिटिव्ह नंबर बिकॉज व्हरायन्स आर ऑलवेज पॉझिटिव्ह. आणि कारण व्हरायन्स हे नेहमी पॉझिटिव्ह असतात. हे कोणाच तरी स्क्वेअर असतात, स्क्वेअर रूट नसतो, हा स्क्वेअर असतो. म्हणून या ठिकाणी व्हरायन्स पॉझिटिव्ह येतं असं अपेक्षित आहे. सिन्स एफ टेस्ट बेस्ड ऑन द रेशिओ ऑफ टू व्हरायन्सेस. म्हणजे एफ टेस्ट मध्ये आपण काय करता, दोन व्हरायन्सचा रेशिओ करता. एक व्हरायन्स वर लिहिता, एक व्हरायन्स खाली लिहिता. मीन्स वी आर टेकिंग जस्ट द रेशिओ इन बिटवीन द टू व्हरायन्सेस. मीन्स द रेशिओ ऑफ द लार्जर व्हरायन्स विथ द स्मॉलर व्हरायन्स. इट इज नोन ॲज द व्हरायन्स रेशिओ टेस्ट. यालाच व्हरायन्स रेशिओ टेस्ट देखील असं म्हणतात. The equation for comparing two variances with the F test is असं लिहिलं जातं. F is equal to S1 square. S1 म्हणजे पहिलं सॅम्पल असं आपण म्हणू, त्याचं स्क्वेअर म्हणजे व्हरायन्स. S म्हणजे स्टँडर्ड डेव्हिएशन आणि त्याचा स्क्वेअर म्हणजे व्हरायन्स डिवायडेड बाय दुसऱ्या सॅम्पलचं व्हरायन्स. हे जरी फर्स्ट सॅम्पल सेकंड सॅम्पल जरी असं म्हणालो मी तरी अर्थ याचा असा आहे, फर्स्ट 1 आणि 2 नाही. तर सुरुवातीला S1 चा अर्थ लार्जर आणि S2 चा अर्थ स्मॉलर. सो वेअर S1 स्क्वेअर इज इक्वल टू लार्जर व्हरायन्स द स्क्वेअर ऑफ द लार्जर स्टँडर्ड डेव्हिएशन. ते कसं काढायचं, आपण जे स्टँडर्ड डेव्हिएशन काढायची पद्धत आहे ना, त्या स्टँडर्ड डेव्हिएशनच्या फॉर्म्युल्यामधून रूट काढून टाकायचा म्हणजे त्याचा स्क्वेअर येतो. त्याचा स्क्वेअर येतो म्हणजेच व्हरायन्स आहे. स्टँडर्ड डेव्हिएशनचा स्क्वेअर मीन्स व्हरायन्स. इट मीन्स whatever root is there in formula of standard deviation, when we delete then automatically it is the square of the standard deviation. म्हणून या ठिकाणी या फॉर्म्युल्यामध्ये स्क्वेअर रूट नाही का, स्क्वेअर केला आपण म्हणजेच व्हरायन्स काढलं. वी आर नॉट गोइंग टू फाइंड द स्टँडर्ड डेव्हिएशन, वी आर गोइंग टू फाइंड द स्क्वेअर. ओके, नेक्स्ट S2 स्क्वेअर. आता या ठिकाणी बी दोन आहेत ना आपल्याकडे दोन व्हरायन्स असणार. दोन व्हरायन्स मधला मोठा व्हरायन्स वर लिहायचा, छोटा व्हरायन्स खाली लिहायचा न सांगता. ते कोण्या तुम्हाला सॅम्पलला 1 म्हणलात आणि कोणत्या सॅम्पलला 2 म्हणलात, दॅट इज नॉट इम्पॉर्टंट. ऍट द टाइम ऑफ रायटिंग ऍट लास्ट, फर्स्टली यू पुट द लार्जर व्हॅल्यू फर्स्ट, देन डिवाइड बाय द स्मॉलर व्हॅल्यू अँड नथिंग एल्स. ओके, assumptions आहे. assumption काय आहे, युअर पॉप्युलेशन मस्ट बी अप्रॉक्सिमेटली नॉर्मल डिस्ट्रीब्युशन. ठीक आहे, नॉर्मल डिस्ट्रीब्युशन असावा. नंबर 2, द सॅम्पल्स मस्ट बी इंडिपेंडेंट इव्हेंट्स. प्रत्येक सॅम्पल इंडिपेंडेंट असावेत, बरोबर आहे. नंबर 3, द लार्जर व्हरायन्स शुड ऑलवेज गो इन द न्यूमेरेटर. वर लिहायचं ऍट द टॉप. टू द फोर्स दॅट द टेस्ट इन टू राईट टेल्ड टेस्ट किंवा राईट टेल्ड टेस्ट आर इजियर टू कॅल्क्युलेट. If you are given standard deviations, they must be squared to get the variances. जर तुम्हाला स्टँडर्ड डेव्हिएशन दिला असेल तर त्याचं स्क्वेअर करून घ्या तर व्हरायन्स येतं. If your degrees of freedom aren't listed in the F table, use the larger critical value अशा अर्थाने म्हटलं. असू द्या, त्याची काही गरज नाही आपल्याला. Steps in applying F test. If you are given standard deviations, जर तुम्हाला स्टँडर्ड डेव्हिएशन दिले असतील प्रॉब्लेम मध्ये, go to the step second. If you are given variances to compare, go to the step 3. जर तुम्हाला ऑलरेडी व्हरायन्सेस दिल्या असतील तर दुसऱ्या स्टेपला जा. जर स्टँडर्ड डेव्हिएशन दिल्या असतील तर दुसऱ्या स्टेपला जा. दिले असतील तर पहिल्या स्टेपला काम करा अशा अर्थाने. ओके. स्टेप 2. स्क्वेअर बोथ स्टँडर्ड डेव्हिएशन्स टू गेट द व्हरायन्सेस. जर स्टँडर्ड डेव्हिएशन दिले असतील तर त्याचं व्हरायन्स करून घ्या, स्क्वेअर करून घ्या. मग S1 दिला असेल त्याला सिम्बॉलिकली सिग्मा 1 म्हणतात. S2 म्हणजे सिग्मा 2 असं देखील म्हणतात. त्याचा स्क्वेअर करा. स्क्वेअर करा म्हणजे S1 स्क्वेअर आणि S2 स्क्वेअर असं करा. तो उत्तर त्या ठिकाणी कॅल्क्युलेट करून दाखवलेला आहे 118.81 आणि हे आहे पहिल्याचा स्क्वेअर आहे 92.16, दुसऱ्याचा स्क्वेअर आहे 118.81. म्हणजे तुम्हाला स्टँडर्ड डेव्हिएशन दिलं. पहिलं आहे 9.6, दुसरा स्टँडर्ड डेव्हिएशन आहे 10.9. या दोघाचा स्क्वेअर केला तर तुम्हाला आलेला आहे 92.16 आणि 118.81. हे दोन आहेत व्हरायन्सेस. नेक्स्ट स्टेप. टेक द लार्जेस्ट व्हरायन्स. पहिल्यांदा लार्जेस्ट घ्या. आता कोणतं मोठं आहे दोघांपैकी? S1 स्क्वेअर मोठा आहे का S2 स्क्वेअर मोठा आहे? तर S2 स्क्वेअर मोठा आहे. हे 118 वाला मोठा आहे, ते अगोदर घ्या आणि नंतर डिव्हिजन मध्ये 92.16 घ्या असं म्हटलेलं आहे. काय म्हटलेलं आहे? टेक द लार्जेस्ट व्हरायन्स and divide it by the smallest variance to get the F value. In the above example, the largest variance is 118.81 and the smallest variance is 92.16. Then the F value will come 118 divided by 81. 118.81 divided by 92.16. The answer is 1.289. Now step 4. Find your degrees of freedom. Degrees of freedom किती आहे बघा. Degrees of freedom is your sample size minus 1. Degrees of freedom चा अर्थच असा आहे सॅम्पल साइज मधून n वजा करणे. Also you have 2 samples. त्यामुळं 2 सॅम्पल असल्यामुळं n1 चं वेगळं येईल आणि n2 चं वेगळं येईल. 2 व्हरायन्स आहेत आणि 2 व्हरायन्स साठी 2 डिग्रीज ऑफ फ्रीडम येतील. You will have 2 degrees of freedom, one for numerator and one for denominator. लक्षामध्ये म्हणजे 2 तुम्हाला सॅम्पल आहेत, ज्याचं जे असेल त्याचं त्याला वापरायचं आहे. स्टेप 5. Look the table value of F for 2 degrees of freedom, one of the numerator and one of the denominator. आता या ठिकाणी दोघांच्या डिग्रीज ऑफ फ्रीडम बघा आधी. म्हणजे वर जे लिहितो तर न्यूमरेटर आहे, जे आपण डिवाइड मध्ये लिहितो ते डिनॉमिनेटर आहे. या वरच्यांना V1 घ्यायचं आणि दुसऱ्यांना V2 म्हणायचं. परंतु असं असलं तरी तुम्ही जे काही लार्जेस्ट व्हरायन्स वर लिहिता त्याची डिग्रीज ऑफ फ्रीडम ते लक्षात घ्या आणि स्मॉलेस्ट खाली लिहिता त्याची डिग्रीज ऑफ फ्रीडम म्हणजे V2 आहे, बाकी काही नाही. नेक्स्ट. Step 6. Compare the calculated value of F with the table value. आपण जे करता तेच आहे. If the calculated value of F is less than the table value of F, then the null hypothesis will be accepted and vice versa. सेम आहे आपण जे करता ते. आता या ठिकाणी हे झाल्यानंतर आपण काय करूया, याचा एखादा प्रॉब्लेम बघू. प्रॉब्लेम F test. Problem number 1. Okay. Problem number 1 on F test. Two random samples were drawn from two normal population and their values are A सॅम्पलच्या व्हॅल्यू आहेत, हे B सॅम्पलच्या व्हॅल्यू आहेत आणि टेस्ट whether the two populations have the same variance at 5% level of significance. The table value is already given. V1 म्हणजे 10, V2 म्हणजे 8 म्हटलं आणि उत्तर आहे 3.36 कम्पेअर करण्यासाठी. हा प्रॉब्लेम आहे. आता दोघांचे व्हरायन्स काढायचा असतो. पहिल्यांदा हायपोथेसिस. Let us take the null hypothesis that the two populations have the same variance. आता there is no significant difference म्हणा किंवा the variance of the two populations have किंवा same variance म्हणा किंवा there is no significant difference between the variance of these two populations असं म्हणा चालतं. आपला F टेस्टचा फॉर्म्युला लिहा. लार्जर व्हरायन्स डिवायडेड बाय स्मॉलर व्हरायन्स आणि लार्जर व्हरायन्स आणि स्मॉलर व्हरायन्स म्हणजे तुम्ही जे स्टँडर्ड डेव्हिएशन काढलात त्याचं रूट फक्त काढून टाकायचं. जे स्टँडर्ड डेव्हिएशन तुम्ही काढलात त्याचं रूट काढा, दोघांचं इंडिपेंडेंट काढा आणि फॉर्म्युला आपल्याला दिलेला आहे. दोघांचं स्क्वेअर डेव्हिएशन मधून सम करून त्याला डिग्री ऑफ फ्रीडमने डिवाइड केल्यानंतर ते तुमचं व्हरायन्स येतं. लार्जर म्हणजे उत्तर मोठं ज्याचं आहे ते लार्जर, म्हणून लार्जर मध्ये खालचंही S2 किंवा S1 दोन्हीपैकी कुठलंही वर येऊ शकते आणि दोन्हीपैकी कुठलंही खाली जाऊ शकते. लॉजिक असं आहे, लार्जर व्हरायन्स यू पुट फर्स्ट ॲज अ न्युमरेटर अँड स्मॉलर व्हरायन्स यू शुड पुट ॲज अ डिनॉमिनेटर अँड नथिंग एल्स. Calculation of two means, two variances and F value. आता एक लक्षात घ्या, आपल्याला दोन व्हरायन्सेस काढायचे आहेत आणि व्हरायन्सेस काढण्यासाठी मीन काढावं लागतं. It is not part of the mean to calculate mean of the F value, but without having mean we are unable to find the standard deviations or variances, because standard deviation means nothing but when we are going to take the deviations from actual mean and then we are going to square it and taking sum and dividing by number of items or degrees of freedom here, and then we are taking square root is called as standard deviation. But here no need to take the square root, only square deviations sum divided by n minus 1 or n1 minus 1 किंवा n2 जे असेल ते. बघा फॉर्म्युला. पुन्हा या ठिकाणी काय केलं आपण, x1 चे आकडे लिहिले, नंतर त्याचं मीन काढलं. 720 आहे, याचं 720 टोटल आले, डिवायडेड बाय टोटल किती आहेत 9 आहेत, तर याचं 80 आलं मीन. दुसऱ्याचं मीन आलं 83 आलं. का सर्वांची बेरीज केली 913 आले, डिवायडेड बाय टोटल 11 आहे, ते डिवाइड बाय 11 केले तर 83 आली. आणि हे 83 आणि हे 80 हे वजा करा कशातून, एक्स व्हॅल्यूज मधून. सो 66 minus 80 minus 14, 67 minus 80 is equal to minus 13, 75 minus 80 is equal to minus 5, 76 minus 80 is equal to minus 4, 82 minus 80 is equal to 2, 84 minus 80 4, 88 minus 8 80 is equal to 8, 90 minus 80 is equal to 10, 92 minus 80 is equal to 12. These are the square deviation. या square deviation ला square करा. Sorry, these are the deviations from actual mean. आणि या these are deviations from actual mean ला square करा. Square केलं नंतर sum करा. Same for sample 2 ला करा. ओके. आणि मग रूट वापरायचं नाही. रूट वापरलं तर ते स्टँडर्ड डेव्हिएशन होते. आपल्याला इथं स्टँडर्ड डेव्हिएशन काढायचं नाही. आपल्याला काय काढायचंय, आपल्याला या ठिकाणी काढायचं आहे स्क्वेअर डेव्हिएशन. सॉरी, आपल्याला या ठिकाणी स्क्वेअर ऑफ आपल्याला या ठिकाणी स्क्वेअर ऑफ स्टँडर्ड डेव्हिएशन काढायचं आहे. म्हणजेच रूट निघून गेला बघा, इथला रूट निघाला, इथला रूट निघाला, आणि मग पुट द व्हॅल्यूज इन द फॉर्म्युला अँड गेट द आन्सर. पहिला व्हरायन्स आलं 91.75, दुसरं व्हरायन्स आलं 129.8, 129.8. आपला नियम आहे लार्जर व्हरायन्स वर लिहायचं आणि स्मॉलर व्हरायन्स डिव्हिजन मध्ये लिहायचं, आणि आपण तेच करणार आहोत आता या ठिकाणी. लार्जर व्हरायन्स कोणतं लार्जर आहे? आता खालचं आहे, S2 आहे. मी लिहिताना फॉर्म्युल्यामध्ये लिहिले, का लिहिलं? मी फॉर्म्युल्यामध्ये लिहिताना लिहिलो या ठिकाणी काय, S1 स्क्वेअर डिवायडेड बाय S2 स्क्वेअर असं म्हणालो, परंतु या ठिकाणी S1 स्क्वेअर म्हणजे लार्जर, आणि लार्जर हे आपलं या एक्झाम्पल नुसार S2 आलं आहे, खालचं आहे, म्हणून लार्जर वर लिहिलं मी आणि स्मॉलर खाली लिहिलं. म्हणून इथं फॉर्म्युल्यामध्ये लिहिताना म्हणालो मी लार्जर व्हरायन्स डिवायडेड बाय स्मॉलर व्हरायन्स, आणि लार्जर व्हरायन्स आहे 1298 डिवायडेड बाय स्मॉलर व्हरायन्स आहे 91.75 इज इक्वल टू जे आले तर आलं. नाउ कम्पेअर विथ द टेबल व्हॅल्यू, कम्पेअर विथ द टेबल व्हॅल्यू, आणि ती टेबल व्हॅल्यूशी कम्पेअर केल्यानंतर कॅल्क्युलेटेड व्हॅल्यू ऑफ F 1.4147 is less than the table value of F, which is कमी आहे at 5% level of significance, and the null hypothesis is accepted. It means that the two populations have the same variance. Okay, thank you. आणि मला वाटतं आपण या ठिकाणी थांबायला हरकत नाही. काय अडचण असेल तर विचारा. ओके
सर पीपीटी शेअर करा ना सर प्लीज सर, हे आम्हाला थोडंसं प्रॅक्टिस करायला सोपं होईल.
आताच का हे करू का
सगळे सर
पुन्हा?
आतापर्यंत सगळे पीपीटी शेअर करा सर. आमचा एक ग्रुप आहे म्हणजे स्टुडंट्स, मराठी सरांना दिलं तर त्यांनी आम्हाला देतील सर हे.
ओके ओके ओके, नो डाऊट. आय विल सेंड व्हॉट्सअप काय असेल ते, आणि ते तुम्हाला सर्क्युलेट करतील. ओके
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 सर.
ओके ओके,
काय
थँक्यू.
अडचण नाही सर, एकदम व्यवस्थित झालं सर आजचं लेक्चर एकदम.
नाही नाही,
काहीच
माझं
प्रॉब्लेम.
सिस्टीमच तशी असते, व्यवस्थितच झालं पाहिजे. कारण काय, यू आर लर्निंग अँड आय एम टीचिंग, अँड मस्ट बी देअर लर्निंग अँड टीचिंग, not
Yes,
only
sir.
complete the particular any course or particular any technical binding, because without having passing you are not allowed to go for a next step.
येस सर.
बरोबर आहे?
येस सर, बरोबर.
ओके सर आहेत का?
Hello. Hello.
सर नाहीत
Hello.
का म्हणलं?
Sir,
hello.
sir, is that I'm
सर
audible?
नमस्कार.
I think sir is not there in the
Yeah, yeah.
our meeting. So from the organizer side, I am thankful to Dr. Harish Chandra Patange sir for delivering a nice session on problem based on T-test and F test. I thankful to you sir from all the participant sides and from the organizer side. And I also thankful to Dr. Vishal Marathi sir, research coordinator, and our college principal for giving me such opportunity. Thank you, sir.
Okay, thank you, thank you.
Thank you, sir. Dear participants, we have to wait for the joining the second speaker. So we have to wait for 5 to 10 minutes. हा सर.
हा सचिन, झालं का सरांचं लेक्चर?
हो सर, सरांचं लेक्चर झालं. आता सेकंड लेक्चर म्हणजे थर्ड लेक्चर सर.
ओके ओके. किती वेळ झाला?
एक दोन मिनिट सर.
ओके ओके, नो प्रॉब्लेम.
येस सर.
So friends, today for second lecture, I think Madam will join at sharp 1 p.m. Today we have with us Dr. P.Y. Bhogavkar ma'am. She is my research guide, and she has ex-director of Government Vidarbha Institute of Science and Humanities, Amravati. So, Madam will join very shortly. Till then, if you have any difficulties, please ask. तुम्हाला काही डिफिकल्टी असतील तर विचारू शकता. येस
सर, ते आता मी सरांना सांगितलंय, ते पतंगे सरांना, की त्यांच्याकडे जे पीपीटीज आहेत ना, सरांनी जे आम्हाला शिकवलंय, त्यांनी तुम्हाला शेअर करायला सांगितलं त्यांना.
ओके ओके.
ते थोडंसं टाका, कारण सर ते प्रॉब्लेम्स आहेत ना, थोडंसं प्रॅक्टिस होईल म्हणजे आमची.
चालते. सरांनी मी वाटलंच पुन्हा सरांना फोन करतो आणि तुमच्याकडे घेऊन देतो.
ओके सर. हो सर.
हो. आणखी काही आहेत? 5-1 मिनिट आपण तोपर्यंत वाट पाहू मॅडमचे. आता हे जे लेक्चर आहे, इट इज जस्ट फॉर शेअरिंग ऑफ एक्सपिरियन्स. म्हणजे तुम्हाला या जे 1 ते 2चं लेक्चर आहे, पण तुम्हाला खूप महत्वाचं आहे, कारण इथिक्स वरती मॅडम रिटायर्ड झालेले आहेत, पण मॅडमचं खूप जास्त कमांड आहे सब्जेक्ट वरती आमच्या बॉटनीमध्ये. तर ते महत्वाचं ठरेल तुम्हाला, मॅडमचे एक्सपिरियन्स वगैरे काय आहेत काय त्याचा. त्यामुळं म्हणजे जरी सिलेबसशी रिलेटेड नसलं तरी आपण मध्ये आता याच्यानंतर आणखी 2-3 दिवस आपण मध्ये खूप चांगले जे स्टॉलवर्ड आहेत लोक त्यांच्या फील्डमध्ये, त्यांचं इंटरॅक्शन तुम्हाला घडून आणण्यासाठी आपण हे मध्ये 1-2 लेक्चर्स ठेवलेले आहेत. तर त्याचा फायदा घ्या. सपोज काही असतील मॅडमला प्रश्न विचारू शकता तुम्ही, काही प्रॉब्लेम नाही. आणखी पठाई मैम, आपका क्या प्रॉब्लम था? हेलो
सर. हाँ सर, मेरा ये प्रॉब्लम था कि आपने नहीं बताया था कि एमफिल के लिए आप यूनिवर्सिटी में जाके पूछो कि क्या करना है. तो मेरा फ्रेंड उधर गया था, तो उसने बोला एमफिल को 2 सब्जेक्ट्स एग्जाम्शन है, लेकिन आपको कोर्स वर्क पूरा करने का है. मैं आपको पूछ रही थी ना सर, कोर्स वर्क मेरे को वही लेक्चर का करना है क्या नहीं? तो उधर यूनिवर्सिटी में स्टार्टेड है. यूनिवर्सिटी में मेरे फ्रेंड्स है ना कॉमर्स और आर्ट्स के, तो उनका भी यूनिवर्सिटी के थ्रू ही स्टार्ट हुआ है. तो उनको क्लियरली फर्स्ट डे पे बताया ऑल एमफिल विल बी एग्जाम्प्टेड फ्रॉम 2 पेपर्स, फर्स्ट एंड सेकंड, एंड थर्ड दे हैव टू गिव, बट उनको कोर्स वर्क पूरा करना है. कोर्स वर्क में कोई एग्जामिशन नहीं है कि 1 पेपर करो या 2, ऐसा. वो आपको बताना चाहती थी मैं. और सर आप बोल रहे थे ना यूनिवर्सिटी में कोई एग्जाम सेंटर में कोई कुलकर्णी सर करके हैं, और
पीजी सेक्शन में, पीजी सेक्शन में कुलकर्णी सर.
और दूसरा नाम सर कौन सा बताया था, वो मैं भूल गई थी.
अपने ये होंगे एग्जाम सेक्शन के, क्या नाम है उनका, एग्जाम के जो मेन है, सरोदे सर.
हाँ. तो वो बोली कि अगर मैं जाऊंगी तो फिर मैं पूछ के आऊंगी उनसे भी. लेकिन उन्होंने क्लियरली बोला है, जब ये यूनिवर्सिटी वालों ने कोर्स वर्क स्टार्ट किया तो बोला फर्स्ट डे पे ही हम लोगों को ऐसा बताया है कि आप लोगों को एग्जामिशन है. तो फिर किसी ने कुलकर्णी सर से पूछा, तो मेरा दूसरा फ्रेंड कोई गया, वो लास्ट 2 इयर्स का बैच का है, तो उसने बोला कि अगर एमफिल के मार्कशीट पे लिखेला होएगा तो नहीं होगा. तो बोला सर ऐसा है कि ऐसा थोड़ी होता है कि मार्कशीट पे लिखेला होगा या नहीं, आपके सर्कुलर बोलता है एमफिल, तो एमफिल ही होगा ना सर, ऐसे करके उसने.
बराबर. नहीं मैम, क्या है उसमें बहुत ज्यादा कॉम्प्लिकेशंस है, इसलिए मैंने आपको सीधा इसलिए बोला.
हाँ, वही ना.
2-3 साल पहले यही प्रॉब्लम हमारे साथ आया था और उनको उन्होंने एग्जाम्पशन डिनाई कर दिया. तो इसकी वजह से आप इंडिविजुअल ले जाओ, यानी आपका जो प्रॉब्लम है वो खुद सॉल्व हो जाएगा. नहीं तो हमको पूछने में कुछ भी हर्ज नहीं, लेकिन वो हमको बोल देंगे कि नहीं है, और बाद में आपको प्रॉब्लम आ सकता है.
सर नहीं वो, हाँ सर, मैं वही आपसे शेयर करना चाहती थी कि जो आपने कुलकर्णी बोला ना, तो वो कुलकर्णी सर से बात करने गए, तो कुलकर्णी सर बोल रहे थे कि जो मार्कशीट में लिखेला होएगा तो वो सब्जेक्ट नहीं करना है, तो उसने बोला सर ऐसा थोड़ी होता है, मार्कशीट में तो 5 सब्जेक्ट्स लिखेला है, तो आप 5 सब्जेक्ट्स नहीं लेंगे क्या? बोला कंप्यूटर नहीं लिखेला है तो कंप्यूटर भी देना पड़ेगा। तो वो टाइम पे कंप्यूटर थोड़ी लिखते थे। तो बोला सर सर्कुलर दिखाओ इधर, वो कौन सा किधर लिखेला है कि मार्कशीट में वो होगा। वो बच्चे ने डिफेंड किया उसको कि आप मार्कशीट बोल रहे हो ना, तो आप बताओ ना, सर्कुलर दिखाओ कि कहाँ लिखेला है कि मार्कशीट में जो आएगा वो सब्जेक्ट नहीं देने का है। तो मेरे को उसने ऐसे बताया। तो और फिर अभी जो रिसेंट चल रहा है, तो वो दूसरी फ्रेंड बोल रहे हैं नहीं नहीं 2 ही एग्जामिशन है, उन लोग ने क्लियर हम लोगों को बताया है, फर्स्ट डे पे ही बताया है कि कोर्स वर्क आपको पूरा 21 डेज का करना है, लेकिन पेपर का एग्जामिशन आएगा और आपके एडमिट कार्ड में वो ही पेपर आएगा, एडमिट कार्ड मिलेगा शायद हमको।
ओके।
पेपर का मतलब एग्जाम का एडमिट कार्ड आने वाला है, उसमें आपका वही पेपर
हाँ, नहीं
आएगा।
नहीं, लेकिन अब इसके बाद आपको एग्जाम फॉर्म भरना पड़ेगा, तभी आप वो एडमिट कार्ड आएगा आपका।
हाँ
यानी अभी एडमिशन लिया है तो डायरेक्ट एडमिट कार्ड नहीं आएगा आपका, इसके बाद एक सर्कुलर आएगा।
हाँ,
वो
तो वो
सर्कुलर,
मतलब हाँ
हाँ,
हाँ, मतलब
एग्जाम
उधर
डेट्स
ऐसा
का।
उनको बताया कि उनको एडमिट कार्ड में ही लिख के आएगा, तो वो लोग क्यों देंगे, वो लोग लिखेंगे एमफिल तो एडमिट कार्ड में उनको एक ही पेपर लिख के आएगा, ऐसा बोला था उन लोगों को
नहीं नहीं नहीं
उधर के।
मैम, आपको सब्जेक्ट डालने पड़ेंगे ना, वो जब एग्जाम फॉर्म भरेंगे तो आपको सब्जेक्ट डालने पड़ेगी कि आपको कंप्यूटर देना है या रिसर्च मेथोडोलॉजी
अच्छा
देना है।
अच्छा, ऐसा है
हाँ,
क्या? हाँ, ओके ओके सर,
आप
हमको
जो
पता
सब्जेक्ट
नहीं। हाँ
लिखेंगे वही सब्जेक्ट आपके एडमिट कार्ड पे आएंगे।
ओके, तो हाँ ठीक है सर। हाँ तो मैं समझ गई ना सर, मैं एमफिल के बेसिस पे मैं नीचे थर्ड वाला पेपर एथिक्स ही डालूंगी ना फिर।
हाँ हाँ हाँ,
ऐसा
तो वही लेकिन वो क्या बोल रहे थे कि आपके कंप्यूटर नहीं है अगर आपके मार्क लिस्ट में, तो आपको कंप्यूटर भी लिखना पड़ेगा उसमें।
नहीं सर, वो लड़के, वो जो दूसरा लास्ट ईयर का लड़का था ना, उसने जाके, मैंने बोला ना उसको पूछ के आने के लिए कुलकर्णी सर करके, तो बोला कुलकर्णी सर ने बोला कि मार्कशीट में जो लिखेला होगा उसमें एग्जाम्पशन आएगा। तो वो लड़के ने बोला कि मार्कशीट में सर 5 सब्जेक्ट्स हैं, रिसर्च मेथोडोलॉजी के साथ कोर्स वर्क 1, कोर्स वर्क 2, फिर कोर्स वर्क क्यों करवा रहे हो हम लोगों से।
हाँ
तो ऐसा उसने डिफेंड किया कि आप दिखाओ ना सर्कुलर किधर लिखेला है कि मार्कशीट के हिसाब से आपको पेपर देने का है। ऐसा
हाँ
मैं आपको
हाँ
वो
हाँ
शेयर कर रही हूँ, ऐसा बता रही हूँ।
हाँ,
वो
ओके
लड़की ने
ओके।
उसको बताया कुलकर्णी को, कुलकर्णी सर को, कि सर आप जो आप बोल रहे हो वो आप कैसे बोल रहे हो, आपका सर्कुलर में तो लिखेला नहीं है कि मार्कशीट पे आएगा, मार्कशीट पे तो 5 लिखेला है, कोर्स वर्क 1, कोर्स वर्क 2, हमने तो 2 कोर्स वर्क किया है, फिर ये कोर्स वर्क क्यों करवा रहे हो हमसे। ऐसा सर, एक्चुअली ना मुंबई यूनिवर्सिटी में एमफिल में पूरा एग्जामेशन है पीएचडी वालों को। मेरी फ्रेंड है ना, उसको मुंबई में मिला, तो उसको कोर्स वर्क नहीं करना पड़ा, लेकिन मेरा क्या है, गाइड की वजह से नहीं मिल रहा था मेरे को, तो 2 इयर्स मैंने वेट किया, तो इसलिए फिर मैंने यहाँ स्टार्ट किया। तो
ओके
मुंबई
ओके।
यूनिवर्सिटी तो एमफिल फुल एग्जाम्प्टेड है, नो कोर्स वर्क।
नहीं मैम, ये कैसा है, वो डिपेंड है, यूनिवर्सिटी वाइज
हाँ,
रूल
हर
चेंज होते
यूनिवर्सिटी का अलग
हैं,
अलग है।
अलग अलग।
ऐसे
हाँ
जेजेटीयू में 2 मंथ्स का है, जेजेटीयू का
हाँ
2
हाँ,
मंथ्स का
बराबर।
है सर, मैं उधर भी इंक्वायरी किया।
अमरावती यूनिवर्सिटी में 6 मंथ का है पूरा, कंप्लीट
हाँ,
सिक्स
लेकिन
मंथ्स
जेजेटीयू में 2 मंथ्स का है, लेकिन 2 आवर्स, लेकिन सर
ओके।
कौन करेगा 2 मंथ्स इतना प्रोलॉन्ग, कौन करेगा।
हाँ
हाँ, तो वही हर यूनिवर्सिटी का वो तो सर अलग अलग है, दैट इज आई
अलग
नो,
अलग।
दैट आई नो। हाँ
यस।
यस सर, बट दैट ओनली आई वांटेड टू शेयर विथ यू।
या ओके ओके, आई विल कॉल द मैम, आवर गेस्ट।
Hello.
Hello, ma'am. Good afternoon, ma'am.
Good afternoon. Should I start?
1 minute, ma'am.
Okay.
I will introduce to you to the. Okay. All dear participants, I welcome you all for this afternoon session. Today we have with us Dr. Prabha Yadav Bhogaonkar, my guide, my mentor. She is actually retired professor of botany and ex-director, Government Vidarbha Institute of Science and Humanities, Amravati. She has very huge experience of teaching and research. Particularly, her area of research is angiosperm taxonomy and ethnobotany. Under his guidance, 16 students were awarded for their PhD degrees, and 102 research paper publications. They are published in various national and international journal of high impact factor and high repute. She has published 4 books, particularly with respect to the books. Dr. Bhogaonkar ma'am is a student of well-known taxonomist, Dr. V.N. Naik, who is a well-known taxonomist of India. He has contributed the flora of Marathwada, and madam has also contributed as a co-worker for the flora of Marathwada, along with the Dr. V.N. Naik, sir, with respect to one of the most toughest family of angiosperm, that is Asteraceae family, one group of Asteraceae she is dealing with. Again, she is one of the first Indian to honor with international award, Janki Ammal Medal Award for lifetime work in ethnobotany. Ethnobotany is one of the important subject of botany, which deals with the plants, relationship with plants and human tribal peoples. Ma'am has very lot of work with respect to the tribal peoples and indigenous knowledge of these tribals with respect to the plants. So to honor her work, this award has she has felicitated with this award, that is Dr. Janki Ammal Medal Award for the lifetime work in ethnobotany. She is also a fellow of Indian Association of Angiosperm Taxonomy, one of the renowned institution, institution which is mainly work in for the angiosperm taxonomy. She is also a fellow of Society of Ethnobotanists. She is also honorary president of IAT, Indian Association of Angiosperm Taxonomy Conference, which is held at Trichy in 2014. She is also vice president of Society of Ethnobotanists in last year, 2020. Again, she is founder president of Wildlife and Environmental Conservation Society, WEX, at Amravati. Again, she is also a founder president of Warsaw Ethnic Knowledge Heritage Society, which is just formulated by her, and along with all her research students. Again, she has helped a lot for the identification of plants near about 15 years to the forest department. She has worked with forest department, particularly in Melghat forest area of Amravati region, for vegetation monitoring in Melghat forest area, along with government of Maharashtra forest department. She is also expert committee member of a biodiversity board, Maharashtra, Pune. So friends, this lecture is a treasure for you, because she is a well experienced and she is also well dedicated hardcore taxonomist, hardcore researcher. And as you are the new budding researcher, particularly you are pursuing for your PhD degree, so it is very much essential for you to just listen to Dr. Bhogaonkar ma'am. So I welcome on behalf of all the participants, on behalf of Research Committee, NES Science College Nanded, and from behalf of Principal Science College Nanded, I welcome Dr. Bhogaonkar ma'am. And now I request Dr. Bhogaonkar ma'am, please start her lecture, ma'am.
Thank you, Vishal. And good noon, everybody present here. Talking about ethics of research and publications is really interesting, but at the same time, it is difficult also, because these are what we call as the moral values. And it's difficult to talk about the moral values and more difficult to practice them. Now here for pre-PhD course, I think students of all subjects must be participating. However, I belong to botany, and that too, my experience is restricted to angiosperm taxonomy and ethnobotany. You can say a narrow specialization. Naturally, whatever I shall be talking will be about the research and publication ethics related to these topics mainly. And many of you may not find it interesting. So please excuse me if you don't find that thing more interesting. Because what happens, we are well connected with our own subjects. And apart from the subject, if someone talks or gives examples of other subjects, since you are not well versed with that subject, you may find it boring. Maybe there are students of physics, students of chemistry, maths, and all that. They will find it somewhat difficult, but I shall try to narrate the things just like a storytelling. Now, when I talk about the PhD or think about the PhDs and research, I remember the time when I was young, I was a student, and that time, very few people were having PhD degrees, which were my teachers and which were the senior professors around. But necessarily, it is not that PhD means a knowledge. Knowledge and PhD degree, they are definitely different. I remember my beloved teacher and head of the department, Professor Mirashi, who was a well-known taxonomist. Professor Dhamdere, a strong physiologist. And when I say professor, they were selected professors, not that lecturer was called professor. So they were professors. And there are some people who did their PhD, but at the end of their tenure. One of the good example is my senior teacher, Dr. Dhore, who did his PhD nearly at the end of his tenure, but as self-guide. Now you can understand the importance of becoming a self-guide. You must be a person knowing everything or much of the subject. Then I remember one more professor from my institute, Professor Bhatnagar, who was not lecturer, I can say, he was head of the Department of English and a very well-known poet. His poems were taught in university syllabi of some of the American universities and all that, but he was not PhD. He produced somewhat 8 students, PhD students, and then lastly, he himself did the PhD. When we felicitated him, he said that PhD is not the beginning, it is the culmination of your knowledge. And now the things are totally changed in reverse direction. PhD is the beginning of now your research or gaining knowledge. And their PhD was culmination of knowledge. Before his PhD, most interesting thing that before his PhD, he was awarded DLIT by one of the American universities. So please let me say, though it is somewhat what I say, that most of the students are doing their PhD because of practical needs. Now, for getting job, they ask whether you are having a degree, PhD degree. So you have to do it, not because you have passion about the subject, but this is of course the first stage of learning the subject. And what happens when it comes down to any degree only? Many malpractices, fraudulent methods, they enter the field. And here also this has happened. This is why now we want to talk about the ethics of research and publications. I think this is really a sorry state of affairs that we are to talk about the ethics of research and publications. Now, these days, we can say even academics has become much more competitive. Today's world say as is very competitive. Even the academics has become more competitive. In recent years, say last 12 years, scientists, scholars, policymakers, and even members of the society, they are finding that these methods which are followed for the research or whatever is the outcome of the research is many a times a threat to scientific integrity, such as fraudulent academic research. You must be knowing, you must have heard many things about it. Then plagiarism. What is plagiarism? Just stating that this is my own work when it is not. It is a theft. These things, they happen. Manipulation of data is very common. Suppression of data. If you are getting negative results, don't show. If you are getting positive, then only show. But in fact, if you are getting a negative research, then there is an opening, there is a new way ahead to think of why you are getting negative results. What is the reason behind it? Whether this phenomena on which you are working is also working in different ways. That must be thought about. Also, much common is the misuse of research funds and all that. That is why we should know about the ethics of the research, because you are going, I think, to do the research not only for PhD, but you will continue. Now, what is the literal meaning of ethics? The literal meaning is study of morality's effect on your conduct, or study of moral standards and how they affect the conduct. In research, conduct is very important. It is, we can say, actually is a set of regulations to be followed for the betterment of the cause. And here, betterment of the cause is what we say is the research. Basically, you or me want to understand why we are doing the research. You should not think research only in terms of the degree of PhD. Getting PhD degree is the beginning of your research. You are just entering. I will give a very simple example to you. If someone learns to read, he doesn't become a reader. Vachak, what we call in Marathi. To become a Vachak, what you need? You need a passion about literature or any type of books, isn't it? And in broader sense also, like reading, research should become your passion. Then only you are a researcher. This you may find somewhat out of way because this is just beginning for you. But from the beginning, आपण जे म्हणतो की संस्कार हे खूप सुरुवातीपासून व्हायला हवेत. हा पहिला संस्कार तुमच्यावर होणं आवश्यक आहे की PhD is the beginning of your research career and not the end of research career. Now, when we are talking about the research ethics, the three major aspects of this research are important. And what are these three aspects? These three aspects are, first is the expansion of human knowledge of the world. So this is applicable to all the subjects. It is something universal about the research. Whatever you are doing, whatever you are your results, they should be addition to the existing knowledge or existing information. I will give a simple example of floristics. Suppose you want to study the flora of a particular region. Then first thing is that you should be sure that at least 20 years lapse is there in the previous work and your work that is going to start. Why? If it's a small period, you won't be adding any new information to the existing one. 20 years is quite a sufficient period that plant flora gets affected in that period. Some considerable changes might have occurred, and one should more focus on the herbaceous flora because it is more, you can say, more fragile, and you may get there some invasions, some environment, some, you can say, new species might have entered, some species might have become extinct because of some environmental changes also. Some modifications, mutations might have occurred in these herbaceous plants because of the climatic changes. And analytical part of the study is more important to precisely understand the effects of microclimatic changes, also human interference. Only enumerating species is not sufficient. That will not add to the information. The information also should have maybe some few species, 2 or 3 or more, you may be reporting newly from the area, but that is not sufficient. The reason behind these invasion of these species, occurrence of these species, or maybe the changes that are occurring in the existing species, they should be explained with very clarity. There you need to understand what is going to affect the flora in different parts, considering the ecology. And also not only ecology, also the human changes, or you can say the human role of human beings in that particular natural area. Second aspect, major aspect of research is deepening of understanding of its aspects. Now, if you consider any topic for the research, you will find that there are so many angles to a particular aspect. Now, if I say that I am going to do the ethnobotany of Medghat region, so ethnobotany is a broad term. There are several aspects of this ethnobotany. If I simply go there, ask the people about what are the plants growing here, how they are used, or what are the uses of these plants, it is not sufficient. That is why we say that cultural study of ethnobotany or any tribe is also important. What is the culture? It is a style of life that has established over the generations together. Certain things, they are preserved in terms of customs. Certain things, they are preserved in terms of myths and beliefs and all that. But everything is related to their knowledge about and their understanding about the plants. Simply asking them how you use them is not sufficient. That is why cultural study is important in ethnobotanical studies. This is one of the aspects of ethnobotanical study. What is another aspect? Another aspect is ethnomedicine. Still another aspect is the wild edibles. Still another aspect is NTFPs that occur in that particular region and on which the economy, the livelihood of these tribals is dependent. And these is to be studied considering the entire social structure of that particular tribe. Only reporting 100 medicinal plants is not an ethnobotanical study. Whatever subject you are studying, you have to look from different angles of your subject, and you will find that this study then will much enrich the existing knowledge of the subject. Another thing which you should keep in mind, this is an what we say eternal truth, that as knowledge expands, it is more likely that something that was accepted as a fact at a given point of time needs modification or maybe even rejection. Not only modification, even rejection. That is why I say that negative results also are important. They are giving you another different clues. In science, particularly, what is science? It is a continuous journey towards the ultimate truth. Journey. We, at any point, we can't say that we have reached the ultimate truth. It is not possible. And that is why do not hesitate to say your findings, even if they are not in line with the present theories or present concepts. You can put the different concepts. Maybe they are acceptable to others, or they may not be acceptable to others. Don't care about it. But what you have got must be put forth very boldly. The third aspect of research is enhancement of human ability to exploit the knowledge for achievement of goals vital to humanity or having social merit. Ultimately, any research we want to work for the betterment of human society, isn't it? Whether it is a chemistry, whether it is a physics, whether it's a mathematics, or whether it's a biology. Biology is more nearer because it is more nearer to the life. Now, again, I will give another example from ethnobotany itself. Now, ethnobotanical studies, many people, they feel that ethnobotany is only a documentation. It is not so. And even if you are documenting the things, there are various angles which you need to search for documentation also. Whatever is done by the previous workers is only your guideline. It is not the path on which you have to walk compulsorily. So search the new avenues, and these new avenues will lead definitely to the enhancement of our knowledge. Now, in ethnobotany, especially ethnomedicine, if we turn to the experimentation to understand how these medicines, tribal medicines, ethnic medicines, they work, I'm sure a day will come when we will achieve our goal to provide better and cheaper treatments for various human ailments, which is most important. Even today, you know that our medical services, they are not sufficient to treat everybody, to give everybody healthy life. We say we are lakhs of doctors. And of course, when I am talking about the doctor, it is not only the allopathy. every branch is important. The only thing is that every branch is to be supported by the research. Actually, allopathy is the only branch which is extensively supported by the research. But here also you will find one thing. Maybe today's medicine becomes non-persistent tomorrow. It is replaced by some other medicine. Why? Because it is a journey to prepare the best medicine, most effective medicine, the medicine which will not cause the side effects or to lessen the side effects and all that. This is how the experimental studies, they lead you to the betterment of the human society, enhancement of knowledge. And why this enhancement? What for this enhancement of knowledge? This enhancement of knowledge is just for the betterment of human lives. This is the important aspect, third important aspect of research. Also, some of the responsibilities are to be accepted by the researchers. Every scientific research or experiment which is conducted by a scientist becomes his total responsibility, because then you are talking about the results. And when you are talking about the results of your experiments, you are taking the entire responsibility of that experimentation. And when particularly these results are with regards to the human health, human lives, its direct effect on physical and mental health, welfare, dignity, and liberty of the people, then we have to consider it. Even in chemistry, such things happen. I'll tell you, very simple example I'll put forth. We all wear different types of clothing, isn't it? We have our choices of colors and shades. Now, how these color shades, they come? They are from the textile dyes. A time has gone of natural dyes because they are too costly, too expensive, still preferred by few, but they are very, very expensive. And therefore, what is the cheaper thing is the chemical textile dyes. Clothing definitely is the basic human necessity, but the amount of water pollution it causes, amount of air pollution it causes is very bad. Sometimes I just remember one film I have seen on National Geographic. It was about these textile industries from Indonesia. They are causing such a tremendous pollution, water pollution, that some of the areas are having only extremely polluted water, and people are compelled to utilize this water only. Children are playing in these hazardous chemicals. People are washing their clothing, their utensils in these chemicals, and just somewhat far away from this, at small distance, there are the water resources they are using to drink. Definitely, these water resources are also extremely polluted. These industries, I think, care least about the human dignity of societies living in the vicinity. You have to respect, ultimately, we have to respect the human life. Such researchers may provide ultimately wealth, But it is not the only thing which we require, not only wealth, but also the health. If human society doesn't survive, for what and for whom you are doing all these things? These industries, they on a large scale are affecting the public health of the populations, which we can say do not have any voice. What is our need, or what should be the fashion? मला हे मराठीत जास्त चांगलं सांगता येईल. गरजेपुरते कपडे असणं, वापरणं ही आता फॅशन बनायला हवी. कपाटाच्या कपाट भरून कपडे आहेत आणि दार उघडलं की खाली पडतायत घड्या, हे नको. त्याची गरज नाही आपल्याला. आपल्याला जी गरज आहे ती फक्त गरज भागवणे ही आता फॅशन बनण्याची गरज बनलेली आहे. आणि आपल्याला आय वुड लाईक टू टेल यू वन मोर थिंग, दॅट इन अमरावती there was planning for the Asia's biggest chemical zone. When myself and my friend Milind Champanerkar, we tried to get the information about this, we found that very, very hazardous industries, chemical industries are coming to Amravati. They would have tremendously affected all the water resources, the entire orange crops that grows in the Vidarbha, which is really a prestigious crop for Vidarbha. And also definitely the water pollution, air pollution would have affected the human life. And with all our strength, we pursued against the chemical zone much more legally, much more scientifically. There was no andolan for that. No andolan. There was a small group of us, 10 to 15 people, who were legally fighting for that. And ultimately, we won. But the decision was really wise, so that no unrest was created. The decision said that these are the most hazardous industries. They were enlisted by us, enumerated by us, and only those enlisted industries were banned, not the good industries. So why I could do that? Because my research taught me to care for the human life and to respect for nature. This is what we expect from a knowledgeable person. Knowledge can be from any field, but when you really gain the knowledge, you really develop the respect about the human life and also about the nature. Now, I think I'm going somewhat more general. Some norms of scientific research should govern our projects. There is an old paper of 1973 by Robert Morton, and what he is called prominent sociologist of science. Why sociologist of science? Because he cared more about the society and social structure. Whatever he interpreted was for the betterment of the society, because the center of his study was human society. Later on, of course, many research papers have appeared discussing about the norms. But even today, Robert Morton's norms of scientific study, they are accepted to a large extent. And what are these norms? In very short, I will try to complete my lecture. Communalism. What is communalism? Now, today, if I utter this word communalism, it will bring to your mind all the recent violations, communal violations, isn't it? But the commune is the society. Commune should be at the center of your research. And what is important? Do not hide any data from this commune or from the society. Your research should be open to all. Do not hide the results. If anyone is not willing to show his thesis to me, I feel something is wrong with him. Either he has not followed the proper methodology, either he has not worked the manner he has to, or he has something to hide which is somewhat dangerous for the society. So our research should reach the society, should reach the people. I always say to my students that publish books on your papers, on your thesis. Publish popular articles based on your thesis. Why? Because the main aim is to reach the people, to reach the society. And these days, most of the theses are on Shodha Ganga, which is an open access to know the research data. This also helps to avoid the repetitive research topics and restrict the data thefts. One very bitter example is there, which I have experienced in my life. I'll share it to you. Because sometimes the people which we feel are respected, they also do this. There was one of the Senate member when I was in Senate in Amravati University, about whom we had a doubt that his research is not genuine. On the basis of his degree, he was there in Senate. So there was a small group of 4 of us, and we tried to hunt the research content. And we found that original thesis from which the matter was copied. We put it in the Senate House and his membership was cancelled. We were not interested in cancelling anyone's membership, but this type of work sets very wrong notions about the research, very wrong practices about the research. This should never be done. If you are not interested in research, plainly refuse to do PhD. Go for NET, SET. That also will give you entry in the in your subject as lecturer. But research is something which has many different angles, like what I can say, the godly notions. We say knowledge is God, isn't it? And these godly notions related to research, which is a knowledge base, they must be respected, they must be followed in our life. So never go for any, say, manipulation or any research theft that way. Whatever you are putting in your research should be most original one. Then another point Robert Morton he has given is universalism. This universalism, I think today we have achieved this in many ways. Anyone from any race, any caste, any gender, any nationality, any culture can work as a research student. It was in old times that in certain subjects, ladies were not allowed. Now this hindrance also is not there now. These bars are not even, not today. Anyone can enter in any field of the research. Then another thing is that disinterestedness. He describes it as disinterestedness. What is disinterestedness? You should not be interested in the things around you which are political or which are personal. When you are aloof from these points, if I am working with someone and I have bias about his personal life, that reflects in my research. If I am working with some group which has some different political alliances, different political philosophy, it may affect my work. You should not allow this type of interference of your mind in your research work. So when you are working, work or think only about your research problem. Do not think about any, or do not allow any personal bias or political bias to affect the work, when especially it is a teamwork. When you are personally working, no problem. But when it is a teamwork, this we should keep in mind. And these days, collaborative works or teamworks, they are more important, because when we personally are doing alone any research, we can tackle only one point, only one angle. But collaboration leads to study the research topic from various angles, and what I can say, it helps us to reach the ultimate truth. Then organized skepticism. What is organized skepticism? This is again the important point. Your research should be open to criticism. Now for PhD, we have started the open vivas, isn't it? Open vivas can be attended by anyone, and anyone can ask you question about your research. This skepticism or this criticism is very important. In fact, conferences provide us better platform for the criticism. And this criticism tremendously helps to improve your vision and quality of work. Also, many times it helps to find more aspects of the study, more angles of the study, more avenues for your research it will open up. Now, after doing the research, the important thing is to publish, isn't it? Now, I'm not going to talk on the publication ethics. That is again a quite broad topic. And maybe there will be a separate lecture for you on the publication ethics. But certain things are to be followed when you publish your work, maybe only in the form of thesis also. First thing you should bear or you should follow is the fair credit. No research is done by a person alone. It is always a teamwork. Help from many persons results in your ultimate goal. And everyone should be given his due credit. Maybe a very small person helping you to clean your lab, he also should be given the credit. Then respect for physical and intellectual property. Now, in ethnobotanical studies, at least we have to give the credit, because whatever we are doing here is not my credit. I am a mediator to follow this knowledge or to say move this knowledge to the larger sector from a smaller sector. I am a mediator. Who is knowledgeable person here? Who is a resource person here? Is that the tribal person? He is that tribal person who is the source of knowledge. So no thesis should be without mentioning of the list of informants with all details, so that anyone can anytime go to that person and visit and ask him. He may be uneducated, but his knowledge you are utilizing, and therefore for you he must be a respected person. Then there is another thing, social responsibility. The social responsibility, my social responsibility is that I should try to utilize my research for the social welfare, not for my welfare. It ultimately is definitely going to do better for me. It will lead to giving me promotion. It may lead to give me name and fame, many things. But the social responsibility is that it should reach to the society and make something better, better changes, make some better changes in the society, or it may do something better to the society members. This social responsibility should be accepted by everybody. As I said about the water pollution by chemical dyes, they are forgetting the social responsibility. The researchers who are inventing, who are discovering the different dyes, they are forgetting their social responsibility. In case of many industries, there is big or tremendous air pollution, even automobile industry. This air pollution ultimately harms the society. I need to discover, I need to build the things which will cause minimum pollution. This is my social responsibility. If I am working in sociology, then it is my social responsibility to act, to work for betterment of that particular sector of the society for which or from whom I am gaining my knowledge, or for whom I am working, so as to say, for my PhD. To think in larger terms about these social structures or social sectors becomes my responsibility. Then another, particularly in ethnobotany and clinical researches, protecting the privacy and confidentiality of human subjects. When I say that ethnobotany should go experimental, now there are certain diseases, certain experiments where people are ready to help, but they don't want to disclose their identity because there are certain social biases attached to these diseases. At that time, I have to respect their privacy. I must protect their privacy. Even if I am going to present the paper on that particular topic, I have to keep the names of the persons away from my text. This is protecting the privacy and confidentiality in case of human subjects. If the subjects are not human, then the question doesn't arise. Then assigning authorships. Many times it happens that even students, after getting their degree, they publish the papers without mentioning guide's name. That also happens. It should never be there. And not only about the guide, but suppose you are doing a research or particular part of research specifically exclusively in collaboration with one person, then mention his name as author when you are publishing the paper. This we have done in one of our recent papers by Sanjay Satpute and myself. There we have put the third name as author, which is one Vaidu with whom we worked on psoriasis. He has every right to be the author of the paper. This one has to remember that assigning authorships from which we have gained the knowledge is important. Then the last and most important thing is intellectual property. IPRs, you know, these days, isn't it? Intellectual property rights. The best example of sharing this intellectual property is by TBGR, Tropical Botanical Garden and Research Institute, where a team led by Dr. Pushpangadan discovered one medicine, Jeevani, which gives energy and vitality. What happened when they were roaming in the jungles? They observed that the accompanying Adivasis, they were putting in mouth some leaves, and for entire day they were enthusiastic, energetic. They may eat or may not, but the energy was, you can say, energy was not depleted. So after, of course, with a good rapport, Dr. Pushpangadan asked them, what's the raz behind this? And they showed one plant, a small monocot plant, Trichopus zeylanicus. This monogeneric family, Trichopodaceae, where there is a single genus Trichopus, and in India only single species occurs in Kerala, that is Trichopus zeylanicus. And then the team worked for 13 years on that plant, and they made the medicine Jeevani from that, patented. And after getting the money from patent, they constructed roads in that tribal area. They built one hospital there and one school for their children. This is the sharing of intellectual property. Mainly in sociological researches and the field like me, that is ethnobotany. These intellectual property rights, they must be shared, because these tribals, they have the real right over that property. No doubt, myself or say other scientists, they work tirelessly on that, but still the origin of that knowledge is these tribals. They are the preservers and treasurers of this knowledge, and therefore whenever the question of intellectual property arises, they should be given the due justice. Now, I think I have taken the prescribed time. Talking about publication ethics becomes a separate lecture. So I was actually to talk both about the research ethics and publication ethics, but then I felt that it is not possible to cover both the aspects or both the angles in single lecture. Thank you very much. Now, whatever questions you want to ask, you can ask me. You are free to discuss if there is a time.
Okay, thank you very much, ma'am. Now I request all the participants, please ask your queries or any doubts regarding the research work. Please. If you have any doubts, please ask. Yes.
And even not just at this time, they can ask me on WhatsApp or through you, Vishal.
Yeah. Yes, if you have any doubts, please ask. We have 5 minutes. You may ask the questions.
Sir, I want to ask ma'am some general question. Can I?
Yes.
Ma'am, I have heard you about a lot. I was very happy to attend your lecture. Still, I'm very happy to attend your lecture and quite well to listen you, ma'am. But I want to ask you some basic question.
Yes.
At the starting of your research, at the early, you have done your PG and you are starting your work for the publication or some PhD or MPhil. You have some fear at that time or not, or you have just followed the steps and you got the more success in your life? Please share this.
I will tell you, at my time, there was no need of any PhD degree, no any MPhil or nothing like that. I did my PhD or I got the PhD degree at the age of 40.
Yes, okay, ma'am.
And till that time, I had some fixed ideas in my mind. I wanted to work with Dr. Naik and only with Dr. Naik because he was most respected taxonomist for me. Then he said that you work on Inuli. It was his dream that some student should work on Inuli because it is a very difficult tribe. He said that you don't bother about the degree. I said, no, I don't bother because I don't need the degree, but I want to work. So I will work on Inuli. Then when we discussed about the plan, he was basically a morphologist. And I was interested in chemistry. So I said, I shall do the phytochemistry. He said, but you have to do the morphology in all details. I said, yes, I shall do, but also phytochemistry. Then he said that I don't know anything about phytochemistry. I said, it's well, no problem. I'll do it on my own. I did my work in various laboratories of Baroda and NBRI at Lucknow. And I said that, sir, this is the bare facts, that chemical facts I know about my plant. It is not that I know my plants. Now you help me to interpret this information, to use this information to know more about the plants I am working. And that way I completed my research. It was for nothing, no gain. And I'll tell you, even I didn't get the increments because I had crossed that particular stage of that particular level of my pay scale, and I didn't get even a single increment of my PhD. It was simply just for the sake of research done for the sake of research. And I think that is why just after publication of 3-4 papers from my thesis, wherever I went in any university, people used to say, oh, you are that student of Naik who worked on Inuli. So this was that.
Thank you, ma'am. Thank you. Thank you for sharing such information.
Thank you, ma'am. Thank you, ma'am. Yes.
Sir, I want to ask one question to ma'am.
Okay.
Ma'am, basically I am from the botany subject.
Okay.
And presently I am working on some wild vegetables for their anti-cancerous activity.
Okay.
But for that I have conducted one survey from the local areas from my region. So in my PhD thesis, should I keep the name of that respondents?
Definitely.
Okay. Yes, ma'am.
Definitely.
In detail.
You must be working in collaboration with some doctor. If it is
my research
activity.
Not yet.
Then how you are going to decide the anti-cancerous effect of that plant?
From the previous literature survey.
Yes.
From
Just
the previous
literature survey. No, that literature survey, it becomes the secondary information.
Okay.
It is better and it is meaningful if you work in collaboration with some either pharmacy college carrying on experimentation on animals, or in collaboration with some doctor and patient who are already taking this treatment, and just keep the case record so that you will clearly understand whether the plant is efficacious or not. One of my students, Dr. Satpute, has done his work in this way only. We made one team. It was one Vaidu, then his patients. They all, with all written consent, one MBBS doctor and one MD pathologist. This was our team. And then simply we are not entitled to carry out the clinical studies. We are not authorized to do that. But what we simply did, we took the case studies. We took the follow-up of the treatment. And one of the disease, psoriasis, on which we worked, is now, the results are now published, and name of Vaidu is there as an author.
Yes, ma'am.
Secondary data you can use, but there should be some primary efforts from your side.
Okay, yes, ma'am. Definitely I will do that.
Good.
Okay, thank you, ma'am. Any other doubts? Anybody?
सर
Yes.
मला एक म्हणजे जनरल क्वेश्चन विचारायचा होता मॅम ना.
हा, विचारा.
मॅम, म्हणजे आता आपण जो काही रिसर्च करतोय तो सोसायटीसाठी किंवा मग सोसायटीच्या उपयोगासाठी किंवा मग त्या पुढच्या उपयोगाने हे करतोय, बरोबर. मला एक इन जनरल क्वेश्चन विचारायचा होता, म्हणजे तुमचं मत जाणून घ्यायचं होतं की सध्या जी काही कंडिशन आहे रिसर्चच्या बाबतीत भारतामध्ये किंवा मग एकंदरीत सोसायटीमध्ये किंवा मग एकंदरीत या पॉलिटिकल सोसायटीमध्ये, तर त्याच्याबद्दल तुम्हाला काय वाटतं?
आता ऍक्च्युली ह्या रिसर्चला खाली ओढण्याचं काम हे अकॅडमिशियन्सनी जास्त केलेलं आहे. मॅनिप्युलेशन ऑफ डाटा होतो तिथे, मॅनिप्युलेशन ऑफ रिझल्ट होतं तिथे, कॉपिंग ऑफ रिझल्ट होतं तिथे. आणि तुम्हाला जर खरोखर सोसायटीसाठी काय करायचं असेल तर नो पॉलिटिकल पार्टी कॅन हिंडर यू फ्रॉम डुईंग दॅट. कांट बार यू.
ओके. पण एकंदरीत या सगळ्या घोळामुळे म्हणजे एज्युकेशनल क्वालिटी असेल किंवा मग रिसर्चची क्वालिटी असेल ती बरीचशी कमी झालेली आहे.
आपणच हे बाहेरचे फॅक्टर्स इन्व्हाइट केल्यासारखं झालेलं आहे. आपण आपल्या रिसर्चला जर स्टिक टू राहिलो तर काही प्रॉब्लेम येत नाही. तुम्ही विशाल सरांना विचारा. मी तर डायरेक्टर म्हणून पॉलिटिकल लिडरशिपच्या बरोबर पण काम केलं, आणि ते काम करताना तर त्यांनी इतकं अप्रिशिएट केलं की त्यांचे फोन नंबर माझ्याजवळ असायचे, मॅडम अर्ध्या रात्री तुम्हाला गरज पडली तर फोन करा. फक्त तुमचा जो प्रामाणिकपणा आहे तुमच्या कामाबद्दलचा, तुमची जी तळमळ आहे ती त्यांच्यापर्यंत पोहोचायला हवी.
येस मॅम. थँक्यू मॅम.
ओके.
Thank you, ma'am. Any other questions? Okay. Thank you very much, ma'am, for your knowledgeable and informative lecture. Really, our research students get much more benefited from your knowledge. Now, I request Mr. Sachin Sinde for vote of thanks.
Sir, is that I'm audible?
Yes.
Okay, thank you, sir. On the behalf of Research Committee NES Science College Nanded and from all the participants side, I thankful to respected Dr. Prabha Yadav ma'am, a great taxonomist, for your valuable and informative talk on research ethics and intellectual property rights. As you quoted, ma'am, one statement: research is the beginning of research career. PhD is the beginning of research career. So yes, we will try to follow all the research ethics in our research career. Thank you, ma'am, for your informative session. I also thankful to Dr. Vishal Marathe, sir, research convener, and Dr. D.U. Gawai, sir, principal, sir, of NS Science College Nanded, for giving me such an opportunity. Thank you. Thank you very much.
Okay, thank you very much, ma'am. Thank you, Sachin. I really thank to Dr. Bhogaonkar ma'am for sparing valuable time from her busy schedule to interact with our participants. Thank you very much, ma'am. I think it is time to end our meeting. Ma'am, can I end the meeting? Ma'am, Bhogaonkar ma'am.
In the meeting, you can declare the meeting is over.
Okay. With the permission of